Okay, so this is, I would label it as lecture 3 because the first one was, okay. So, so yeah, it appeared now as live again. Okay, let's continue on. So again, RT-PCR is reverse transcriptase polymerase chain reaction. It's more of it falls under the realm of molecular biology, and it's gonna take us a long time if I discuss it to you guys in this forum because we only have several hours left before we end this. But anyway, quantitation of HCV is useful for what? For the determination of the extent of the viremia. Okay, why is it useful? Because we want to see if the patient is capable of surviving the effects or whether or not they require treatment. That's the reason why we need to quantify HCV. All right? What is the purpose of HCV genotyping? This one is another thing that you need to remember. Why? Because certain strains of HCV does not respond to therapy, and therefore we need to know if that particular strain will respond to a particular therapy. Okay? Before we use what? Adjuvants, or what we call adjuvants. We can also use interferons. These are the types of therapies that they use for patients who are infected with viral infections. Interferons is more of like preventing existing cells from being infected by a specific virus. That will be a discussion in an immunology lecture, and I do not want to step on the toes of the lecturer who would be discussing it to you guys. But anyway, let's move on to hepatitis A, Hepadna viridae. Um, Origin of the name again. Uh, most of our lectures right now, most of the slides right now is more of the origin story of many of the viruses because I think it gives a little bit more of it more it gives this lecture a bit more interesting and an interesting take. So hepadna, very easy to remember. Hepatitis plus DNA, hepadna viridae. See, very easy to remember. Right now, what is the virus associated with the hepadna viridae family? Please don't forget that hepadna viridae is your hepatitis B virus. All right, don't be confused with the excess A. All right, hep D hep DNA virus. Okay, so don't be confused. It causes hepatitis B. Now, this virus is a co-infection associated with HBV. Without looking at your notes, this is this is a board exam question. It's a must know. I would like you guys to put a star on this particular slide, highlight this, or do whatever you want as long as you remember it, because this will be asked in the board exam. What is a co-infection associated? Which virus is a co-infection to HBV? Without looking, and I don't want
Hepatitis B
question
po,
mark.
sir.
Hepatitis D. Is it okay? Wonderful, wonderful. But I have to ask Rainiel a question again. Was it advanced reading or Google search?
Advanced reading na po, sir.
All right. Next time you see this question, challenge yourself. Please challenge yourself. Answer the question without looking at this next slide. At the next slide. All right, Rainiel. Thank you and have a wonderful day. Let's move on. Lakas mga call center. All right, let's move on. What is the term used for the infectious particle of Hep B virus, of the hepatitis B virus? Well, it's called as the Dane particle. It's also the other term for this one is your Australian antigen. Wow, Australia, mate. All right, let's move on. Herpes viridae family. There are 8 viruses under this family, but we're going to talk about certain viruses under them. We'll just need you guys to have an overview of what we're going to talk about, and this is what we're going to talk about. Yeah, I know, so much numbers. I know, guys. But let me try to at least give you a little bit of information about each and every one of them. What are the general characteristics of the families? Sorry. What are the general characteristics of the viruses under the hepatitis Herperividae family? Well, they are capable of latent infections. Again, what do you mean by a latent infection? Some members can produce a cytoplastic effect. The immune response is primarily cell mediated. Let's try to dissect what each of these highlighted terms mean. Latent infection. Please discuss it to me quickly. Um, Alyssa, what do you mean by a latent infection? From our previous slide earlier and my previous discussion, what do you mean by a latent infection?
Inactive po.
Inactive? Is that all?
Opo.
Is that all? I feel the answer is a little bit incomplete. Let's try to complete it with Mr. Cesar Makatulan. I really love your name. It's very manly. Cesar, you can type it on the chat, you can type it on the you can unmute yourself. Doesn't matter as long as you're
Hindi
able
po
to
ba
unmute.
Tagalog?
Huh?
Tagalog. Sir, kwan po,
Okay.
yung hindi po siya nagsushow ng symptoms.
Uh-huh. And then, is that all?
Tapos sa later part po siya
Uh-huh.
Cesar, ikaw na.
Cesar, help her out. Medyo nag-nosebleed na si ma'am. Si Alyssa pa si ma'am tuloy. Si Alyssa. Okay, let's try with a different person. Um, Cesar is not in the house right now. Um, let's try with AJ Aquino. AJ, are you there? All right, wonderful. He's not there. All right. Um, yes, yeah, AJ. What do you mean by a latent infection? Oh, late kayo magsagot ng yes po. All right, but I will still add late po siya nagsushow ng symptoms. Very Tagalog. I love it. But the examples be in English. That's the reason why I'm trying my best to speak to you guys in English because the questions might be presented to you in English. But it's okay for you guys to answer in Tagalog. Don't worry. Okay. Um, late po siya nagsho-show ng symptoms. A little incomplete. Please try to complete it for me. Hidden infection, huh? Cesar. Cesar, how hidden is hidden? All right. Dormant. All right. Please explain it to me. Please explain it to me, Charisse. I want you to unmute yourself. I know you have a microphone. Before we move on, Charisse. Charisse.
Sir, yung pagka... Hello po.
Oh, hello. You are
Sir,
you
yung
are now on the
pagkaintindi ko
line,
lang po.
Charisse. Latent infection.
Pagkaintindi ko po,
Uh-huh.
latent infections, parang inactive po siya or nasa body po siya in a dormant po. Parang inactive nga po and hindi po nag-show ng symptoms.
And then kulang. Medyo kulang ng konti lang. I want a little bit more. May konti pa. Konti na lang. Ano pa, Charisse? Wala na? Alright, let me help Charisse out.
Yun yung
It's in the
parang
dormant
incubation.
stage. In a dormant stage. Wala na, Adrian. Huli ka na. Wala na. Nagtampo na ako, Adrian. Adrian, wala na. I feel disappointed with Adrian. Late ka na sumagot. Um, hindi na. Mamaya-maya, after 10 minutes. Ganun na naman ako. Hindi ako petty. Um, Latent infection, just for your information, it's an infection that does not show symptoms, primarily because the virus or the infectious agent is is in a dormant stage. Now, at some point in a person's life, because of external factors, the infection might present itself again, or it might show symptoms. So, yun yung hinahanap kong kulang. Alright? Um, asymptomatic is you have the virus, the virus is active, but your body is strong enough, or your constitution is strong enough to suppress the virus from showing from showing you from making you ill. That's asymptomatic. Latent, the organism, or rather the infectious agent, is dormant, but in the later stages of your life, it might be. Yeah. Yes, Cesar, I know. I know, medyo mahangin. I know, Cesar. I know, Cesar. All right, let's move on.
Static, sir.
Ewan ko sa'yo, Cesar, bahala ka dyan. Let's move on na, Cesar. Hindi ko alam kung anong ibig mong sabihin sa static na yan, Cesar. Hindi ko alam kung chinacharing mo lang kami, Cesar, or what. Napatagalog tuloy ako. My goodness. All right. Let's move on with cytopathic effect. Which organisms are capable of doing cytopathic effect? Basically, what cytopathic effect is, it's capable of transforming the cells again, but it's not more of the cancer form, but rather it's uh, the interaction of the cell to the other cells around it. So that's cytopathic effect. Immune response is primarily cell mediated. What do you mean by immune response is primarily cell mediated? Basically, the immune system is going to rely on the helper T cell, cytotoxic T cell interaction slash memory slash B cell production. No, cytotoxic T cell portion, not the antibody mediated cell response. Okay, so that's the basic information that I need you guys to understand. Now, which subgroup of the hepatitis virus Herpeviridae family causes CPE? This is the one that causes warts. This is what you call. That's the reason why I added cytopathic effect, because they cause warts. All right, they could be in the form of cancer or any other thing. This is the subgroup is the alpha subgroup, which includes herpes simplex virus 1, virus 2, and of course varicella zoster virus. All right, the alpha subgroup. Now, let's move on to the genital infections associated. Now, let's move on to genital infections. Hepatitis, hepatitis, um, hepatitis, herpes simplex rather. Sorry, kanina pa ako ulit ng hepatitis. Ulit na ulit. Favorite kong hepatitis. Let's continue with herpes simplex. Herpes simplex affects which one? What are the genital infections? Herpes labialis and the herpetic whitlow. Well, basically, what these things are is just genital warts. Okay, warts. And I would show a picture, but unfortunately, I had to really remove it last minute because we're gonna show it in YouTube, and it's a little bit. Okay, so not we might not be able to. We might get the video might be taken down. All right. Now, the site of latency for herpes simplex 1 and 2, is the sensory nerve ganglia. It will it will stay on the it will stay on that nerve on its latent stage or its dormant stages, and then the possible clinical conditions that might be associated with HSV1 is aside from genital whitlow and genital warts, or herpes labialis rather, is what you call your gingivostomatitis. So it can cause problems in the mouth. Herpetic keratitis, again, that's another fancy term for a wart, and encephalitis. Why would it cause encephalitis, sir? Because of the fact that it stays in the sensory nerve ganglia. If you're not familiar where the location of your sensory nerve ganglia is, it's actually a link of nerves that's related to your CNS. Okay? So if the virus is capable of replicating in that particular place, it might possibly lead to herpes encephalitis. Okay? So please be careful with who you have sexual intercourse with, because they might have herpes. and they might have herpes simplex 1. And there's a lot of diseases or a lot of conditions that are associated with them. All right? What is VZV? Anybody? Quick lang. Medyo mabilis lang tayo.
Varicella zoster.
All right, pinatawad na kita.
Hiv virus.
All right, thank you, Adrian.
Yeah, thank you,
Pinatawad na
sir.
kita. Thank you so much. Yan ang gusto ko sa iyo, Adrian. Active ka. All right. Wonderful. Wonderful, wonderful. Now let's move on. Varicella zoster. All right. This is varicella zoster virus. Diseases caused by varicella zoster. What are the diseases caused by varicella zoster? Very quick lang without any.
Chicken pox.
Okay, sumusobra ka na kaagad. Sumusobra ka na, Adrian. Saglit lang. Iba naman. Baka sabihin ng mga classmates meron tayong favoritism. I don't have favoritism. Okay? Let's try with Regine Duran who just entered the chat. What are the diseases associated with VZB? She left. Wonderful. Joanna Baltar. She left. Joanna. Shingles. All right. Another one. It's very common in children.
Sir, chicken pox po.
Okay, thank you, Rainiel. I'm not asking you, but thank you so much for interrupting Joanna's brainstorming. Thank you so much. Lovely. Let's move on. Thank you. Thank you from Joanna. Maybe this is a person that might be, maybe Joanna, maybe Rainiel might be, I don't know, romantically related to you because of helping you answer this, because of the help he gave you for answering this question. You might consider it as a romantic gesture, Joanna. Okay. Site of latency for the VZB virus. It's actually now the dorsal root ganglia. Sir naman, ang dami kong kailangang aralanin. Take note, guys, that this slide is actually 551 and we're still at 191, guys. There's a lot more things. And that's just virology, guys. Please don't forget that one. All right? So what's the characteristic shape of the vesicles produced by chickenpox? Another clinical term. I don't know if we might be asking the board exam or it might be presented to you in a case study, but I put it here just in case. It's actually characterized as having rose petal shapes or rose petals with dewdrops. Why does it look like a rose petal? Well, basically it's raised and then the vesicles are slightly clear as opposed to the other ones. Okay, other vesicles that might be associated with other diseases. All right, so they are rose petals with dewdrops, or a rose petal with dewdrops. Now, what's the difference between chickenpox and shingles in terms of clinical presentation? Um, Chickenpox are basically just disseminated lesions, while shingles, which usually occurs in older individuals, they suffer localized lesions. So chickenpox it spreads out, while shingles it's a localized lesion. Actually, chickenpox, for your information, becomes as I mentioned before, it becomes late. It undergoes its latent stages and stays in the dorsal root ganglia, right? What happens is that when the patient becomes immunocompromised, shingles can occur. And I think in the Philippines, people will often say, ay nagka-chickenpox kasi yan, kaya siya nagka-shingles. I think that that's some way of talking about it, but a more scientific way of it is that chickenpox or the VZB virus underwent its latent stage or underwent its latent phase of its infection, and then shingles appeared because the patient has some sort of external factor that reactivated the virus. All right, EBV. What do you mean by EBV? EBV, anyone? What does EBV mean? EBV?
Epstein-Barr virus.
Epstein-Barr virus. Wonderful. Epstein-Barr virus. All right. And what is the disease caused by this virus? Oh, Adrian, sige, nag-raise ka ng hand mo. Adrian, what is the disease caused by Epstein-Barr virus?
Herpes virus. Ano po? Herpes po.
Huh? EBV, Epstein-Barr
Ah,
virus. Virology.
infectious, infectious mononucleosis po.
All right, wonderful. Now, was that a Google search or was that advanced reading?
Pagkakatanda lang po, sir.
All right,
Sa
wonderful.
IS po.
Not to brag, ganun na. Not to brag, sir, but that I answered that based on my memory. Love it. Wonderful. Keep it up. Okay. Um, what happened to my presentation? All right. What happened? Oh, okay. Um, Next, infectious mononucleosis. Another term for infectious mononucleosis. That is the common term or the layman's term for infectious mononucleosis. Do you guys know what's the common term? I already said it. I think I mentioned it before, but I just want you guys to have a little bit of a review. What is the other term for infectious mononucleosis or the layman's term for infectious mononucleosis? Rainiel, advanced reading. Wala. It's probably playing some sort of game again.
Sir,
Ah, what is the other term?
kissing disease po,
All
sir.
right. Thank you, AJ, for answering also with a question mark. Next time, try to guess. It's okay for me if you guess. Don't worry. I will not judge as long as we correct your guesses. If it's a wrong. Thank you, AJ. Have a nice day. All right, let's move on to the next slide. All right. Uh, Next slide. What is the form of IM caused by HIV in HIV infected individuals? This one is a question that was, I think, asked in an ASCP question bank exam. This is ASCP question bank exam. So for your information, it actually causes hairy cell leukoplakia, or what we call oral hairy leukoplakia. Okay, leukoplakia. So it's a common it's one of the questions in the AACP question bank. Again, they seem to forget that we are just responsible for the diagnostic tests, and they ask us pathology questions now sometimes on a rare case-to-case basis. Now, what oncogenic conditions are associated with EBV? Hematology-related questions. Let's try another person from the chat room. Let's try from um, let's try from Alyssa because I think she was raising her hand earlier and I don't want her to feel ignored. All right, Alyssa, what hematologic condition is associated with EBV? Somebody answered already. Sorry, I have to move on with Joanna. Yes, Burkitt's lymphoma. Wonderful. Thank you so much. So this is the reason why I talked earlier about the possible outcomes of a viral infection. It's cell death or activation of oncogenes. All right? Okay? Wonderful. Now, let's move on to another discussion. Yeah, I forgot to mention nasopharyngeal carcinoma. Uh, for us, it's not very common. What I want you guys to remember is that it's associated with Burkitt's lymphoma or Burkitt's leukemia because it might not be asked in the microbiology portion of the board exam, but I'm most certainly sure it will also be asked um, in your hematology examinations. All right, so please be mindful of these things. Site of latency of EBV. Ah, this one is different. This one is different. What is the site of infection for EBV? Latent infection, rather. What is the site of latent infection for EBV or Epstein-Barr virus? Anybody in the chat? You can
Sir,
you. Hmm? Site of latency infection. It's actually just a cell. Again, we have dorsal root ganglia for VZV. We have we have the sensory nerve ganglia for your, somebody answered B cell. Thank you so much, Adrian. Thank you. Rainiel raised her hand, but unfortunately the answer was already given by Adrian. Thank you so much. All right, let's move on to the next slide. Please put a star on this one because this is a must-know question. All right. All right. What is the B cell receptor? Again, a serology question or a microbiology question. May not be asked in microbiology, but it most certainly will be asked in your. If it's not asked in your microbiology, I'm positive that it might be asked in your serology. What is the B cell receptor that binds to EBV? Actually, this was a question in my board exam. If I'm not mistaken, if I'm not, there's so many questions that I've. Yeah, I think this was asked. No, it was asked in my friend's board exam.
Sir?
The B cell receptor. I can hear people from the chat box. CD21, CD3D. Yes. Yes, girl. Somebody answered correctly. Somebody, it's okay also if you answered CD3. That's fine with me. Don't worry about it. Thank you so much for participating. I love the energy that you're giving off. It gives me positive vibes. Love it, guys. All right. CD3D receptor is fine with me. CD3 is also okay because in the exam the form was this one, CD3 only. The choice was CD3 only. Now, I'd like to give the next slide is a little bit more complicated. Let's try to whittle it down into a much more digestible or much more understandable form. How does EBV infection transform into a malignant form? Well, let's start at the first bit. A cell gets infected, the virus gets internalized and undergoes transformation, after which the cell proliferates and EBV genes pass on the progeny cells. What are the progeny cells? Well, of course, that will be the memory B cells. The progenitor EBV DNA remains in the latent stage until reactivation occurs. So the more cellular division happens, the more likely these genes will be spliced together, and therefore the number of chances also, or the statistical chance of getting oncogenes because of the splicing can occur. And reactivation is another way of reactive what if the EBV virus DNA remains in the latent stage until such time reactivation occurs. That's the time that's the time you will get an oncogenic gene. All right. Now, what is the antibody present in patients with infectious mononucleosis? Serology review. Serology review again. I know I'm asking a lot of serology reviews, but it's important also for you guys because we want to connect the dots between the subjects that you're talking about. So what is the antibody present in infectious mononucleosis? Anybody? Anybody in the chat? Advanced reading, I don't care. Heterophile what? Heterophile antibodies, correct. And if somebody will be able to answer this, they will get a prize later. What is the most or the traditional test to detect heterophile antibodies? If somebody is capable of answering that, I will give them a prize later on, sent via Gcash by my mother. Anybody, I will give 10 seconds on the clock.
Spot test po, sir.
Incomplete. Almost there.
Monospot test, sir.
Ah,
Mononuclear spot test.
huh? No. Traditional test. Monospot test. Regine Duran was
Mononuclear.
the one who answered correctly.
Sir,
I'm sorry.
nag-monospot test din ka.
All right. Regine Duran, please email me
Yes po, sir.
yes, please email me your number. I will send it. My mother will send you a 50 peso Gcash award later. All right?
Thank
O, diba? Lakas makawawawi.
Thank
Diba?
you po, sir.
Email me your your email your cell phone number. Hopefully it's a if it's a globe number. If you don't have one, you can have somebody. You can ask somebody else to get claim your prize. All right.
Okay po, sir.
All right. All right. So heterophile antibodies is tested with monospot test. She's correct. And the screening test is actually the monospot test. And then the confirmatory test is Davidson Differential. All right, Davidson Differential. Now, I'm not going to discuss the intricacies of Davidson differentials, but I want you guys to remember sheep red blood cells and guinea pig kidney cells. That's basically what you need for Davidson differential. All right. Now we have another one. What is CMV?
Cytomegalovirus.
Virus.
Ayan, naging active sila nung
Megalovirus.
nagbigay ako ng 50S. Nabigay ako ng ano, my goodness. Sir Marco, naging active sila, no?
Sir, ako po,
Ganyan,
matindi lang po pangangailangan.
ganyan yan, sir. Pag may rewards, active sila.
Oo, I need to motivate the students, apparently. Naging active sila kanina, tahimik eh. I love it,
Gusto yata
people.
nila lagi game show eh.
Oo. Don't worry, there will be another one, but this time, I hope you have studied for the previous lecture. Alright? 50 Gcash yun, oh my God, ano na pwede mo mabili? Parang malaki na ba guys ang 50?
3 days na rin po yun na load.
Sir, 500. Sir, kung 50 reals po yun,
Ay,
500
hindi pwede.
na po sa
Kailangan ko yung
Pilipinas or more.
50. Kailangan ko yung 50 pang load ng Genshin Impact ko. Sorry.
Sir, ako din eh.
Hindi pwede yung 50 SR. 50 pesos lang kasi malaki ang price natin do sa contest ko later. All right? But that prize would be given in on my birthday. O di ba? All right? So next na tayo. Next subject. Next topic na tayo. Cytomegalovirus, yes, that's correct. What disease is caused by CMV? Heterophile negative infectious mononucleosis actually is the term for that one. Why? Because it mimics the clinical symptoms mimics the disease of infectious mononucleosis, and the transmission is actually almost the same. But when you look at the test, it's actually heterophile negative. Okay, the heterophile antibodies are negative. And what are the four infectious stages of CMV? There's actually four: asymptomatic, congenital, and CMV mononucleosis, and CMV reactivation. Because again, since it's under the Herpeviridae family, it is capable of latent infections. All right, and I need you guys to please put a check mark on congenital. Why is it important for you guys to remember the congenital CMV? It is because if it's not gonna be in serology, if it's not gonna be in blood in microbiology, it will be asked in your serology question. It might also be asked in blood banking. The reason being is that it is important as a part of the screening panel for pregnant women. Okay? Especially for blood also for blood donors. We also need to screen for CMV for blood donors. Why? It's dangerous. CMV can actually cause um, birth defects as well as mental retardation in children. So we want to avoid those things. So what are the two populations who are at risk for transmission of CMV, especially with blood buying, or what you call blood buying, unscreened blood. We just wanted to do this and that. That's the reason why we have in MTL um, we have the National Blood Services Act, because there were a lot of people who kept buying blood but they don't screen their donors. And CMV was actually one of the most common disease that has been transmitted in our country before. That's the reason why they have the National Blood Services Act, is because we want to avoid transfusing this one. And aside from transfusion patients, CMV also affects your AIDS patients. Okay? So again, AIDS is a syndrome that occurs because of low CD4 count. AIDS syndrome will occur in patients who are infected with HIV with low CD4 count. Specifically, how many? We'll talk about it later. All right. Which patients require CMV blood culture? Sir, blood culture pala ang CMV? Yes, of course. For virology studies, yes. It's different from your normal bacteriology cultures, but these cultures are observed usually with the electron microscope, a fluorescent microscope, and they are oftentimes requested in hospitals who admit patients with AIDS, who else? Organ transplant patients, and even bone marrow patients. Actually, the best specimen for CMV culture is your buffy coat. You know, the white portion of the blood. Okay? So the white portion of the blood when you centrifuge it, that's the one that we need to use. Okay? So, yeah. So hospitals who admit patients with AIDS, hospitals who do transplantation, who are involved in transplantation medicine, they usually ask this one, because why? We want to make sure that the blood or the tissue sample or the tissues that are being transplanted to the patients are not infected with CMV, because it's rarely tested before in the 90s. It was rarely tested in the 90s, and now it's very very much routine now to test for it. Now, HHV is basically your human herpes virus, um, childhood disease that is caused by HHV6 and HHV7, ruseola. Okay? Another term for your ruseola is what you call exanthema subitum. All right? Not an uncommon question to ask, but I added it here because I remember what's my note for HSV. Yeah. They asked this in Pamet quiz show. In a Pamet quiz show. Maybe some of you might be competing. I don't know. Um, you can go back to the slides if you want to, or you can watch the video again and again. They might ask you about this question. All right. Other term for ruseola, again, exanthem subitum. So ruseola. I guess that's the end of our lecture for. Yeah, I guess that's the end of our virology lecture. Um, I didn't include. Wait, I didn't include HIV because it will. No, there's more. I think there's more. It's just a blank. Yeah, it's just a blank 4 pages because I was trying to update it, and then there were some inconsistencies with my notes. So it's not yet the end of the virology lecture. All right. So malignancy associated with HHSV, Kaposi sarcoma. Now again, you might ask me, sir, you said earlier it's associated also with HIV. Well, yeah, both of them are, but HIV, the AIDS patients would actually get Kaposi sarcoma. What are Kaposi sarcoma basically? They're forms of they start out as small lesions in the skin, and then suddenly it spreads out. Um, I guess because of the malignancy and the ability of the cells to become to multiply, the ability to of the malignancy to uh, infiltrate different body sites. So Kaposi sarcoma will appear like a small mole, and then eventually, after several months or so, it rapidly becomes a large lesion. All right. So HHV, not HHV, HHV8. All right. Not the others, not 6 and 7. All right. I was trying to update it, but the information was inconsistent, so I omitted some of the lectures because I think it would be for advanced an advanced class for virology, not for people who are taking the board exam. All right. Which Herpeviridae family member is associated with non-human primate infections that can cause severe encephalitis in humans. Herpes B virus. So again, another thing that you need to remember is the herpes B virus. Now, let's move on to the family of Ortho Mixovilidae. Again, how many segments are present in the viral genome of Ortho Mixoviruses? There's 8. Please don't forget the other ones. The other ones are segmented as well. But the reason why I repeated this one is because it was asked in a question bank question. It's a question bank. It's a question bank inquiry. All right. What is the shape of the virion of orthomyxoviruses? It's actually spherical shape. All right. The virion, not the not the RNA structure. All right. And what structure surrounds the nucleocapsid of this one? It's actually surrounded by two types of glycoprotein spikes. One is the HA spike or the hemagglutinin spike. The other one is the neuroaminidase spikes. All right, the hemagglutinin spike and the neuroaminidase spike. Um, we have what you call anchor proteins as well, and this is M protein, but it's not to be confused with the M protein in bacteriology, which we will discuss later. Acid prevents mucin production. Now, from this particular glycoprotein spikes, you can you can basically assess pathologic the pathology of a particular virus. Why? Mucoid, mucin production. It affects the respiratory tract. So, what is the importance of HNA in pathogenicity of the orthomyxoviruses? I'll give you a drawing, guys. Um, Neuraminidase breaks the mucin of the host, which is found in the mucus, and then HA binds to sialic acid and allows the absorption of the virus. Okay, so these two things will cause the virus to mucin prevents it to be broken down. Sialic acid binding allows the virus or the viral particle to enter the host cells. All right. Now, under the Orthomyxoviridae family, one is the most important that you need to remember: the influenza virus. All right. So it's the influenza virus, Orthomyxoviridae family. And there are 3 types of influenza viruses, but only 2 are considered as human pathogens, and it's actually influenza B and influenza C. And influenza epidemics are what causes influenza epidemics if the antibody is present, the virus is protective because of antigenic drift. What do I mean by antigenic drift? Basically, what happens is as the cell divides, or rather, as the virus is being produced, it undergoes certain antigenic restructuring. So that's the reason why you need to have an influenza shot every month or so or every season because of antigenic shift. So as to prevent a pandemic. Okay? Now, antigenic drift occurs during viral replication. Yeah, that is the thing that I've already mentioned. Um, Basically, what happens is there will be a mutation in either the hemagglutinin, hemagglutinin glycoprotein or the neuroaminidase genes. Okay, so that's the antigenic shift. All right. So human influenza and animal influenza can actually co-infect each other at the same time. What do you mean by co-infection? For example, a person is infected with swine influenza, and then that person, I don't know, eats the pork products. They will then get infected by the virus, and then the virus will then mutate the cells, and then making human influenza. Okay, that's the reason why we have HN1, HN, H1N1. That's the reason why you also have that's the reason why you have H1 and N1. It's because of these two glycoproteins. Okay, so what that basically means is that hemagglutinin 1, neuraminidase 1, H1N1 virus. That is the reason why you have H1N1 in some of the viruses. Okay, H1N3. So that's how they serotype these viruses. Ortho Mixoviruses, specifically your influenza virus. All right. So hopefully that was thing. So hopefully that sort of information is a little bit enlightening for you guys, because when I was a student, I didn't understand this particular drawing. My professor just gave it to me. We have neuramin. You just need to remember it's neuraminidase and then hemagglutinin. So what's the importance? Well, basically, this will give you the idea that there's a mutation in the neuraminidase. There's a mutation in the hemagglutinin portion. So yeah, okay. So now you understand that clearly, hopefully, guys. Okay. So another term for another thing that we call it is antigenic drift. All right. What are the general categories of the diagnosis of influenza? Well, it's sort of advanced virology, but I'll try to, or advanced diagnostic. It falls under the realms of clinical pathology also, specifically medicine. But we'll try our best to understand each. Viral isolation using viral culture to is one way for us to study this virus. And then another one is the detection of viral proteins, as I mentioned before, the glycoproteins. We can also use detection via RNA PCR. Sir, what's the difference between reverse transcriptase? As I mentioned before, reverse transcriptase is a different form of that one. This one, we're relying on the RNA of the virus. Okay? Um, on reverse transcriptase, we're relying on an enzyme to convert DNA to RNA, so that we can measure how much is the viral load, or how or um, we can detect we can detect how we can detect what particular if there is the presence of minute amounts of the virus. Okay, so that's the reason why that's the difference between those two. All right, and zero diagnosis is another way we can diagnose it. Um, whether it's whether it's determining which hemagglutinin mutation you have, or neuraminidase a person has, or the infection. I'm talking about the infection, or whether or not the patient has influenza or not. All right, let's move on to the importance of the viral proteins. It helps identify what antiviral we're going to use. Yes, there are antivirals that we can use for um, the orthomyxoviruses. All right, specifically, we don't want to use drugs because they have side effects. What we'd rather have is the vaccine, which is given on a seasonal basis, depending on which influenza is common. All right. Now let's move on to the next set of viruses, the Pyromix of viruses. It helps detect which antiviral drug is. Sorry, paramyxoviruses. Let's talk about the differences between paramyxoviruses and orthomyxoviruses. Orthomyxo, based on the name itself, the RNA is segmented, while paramyxo, the RNA is a strand. All right. Remember that orthomyxoviruses have 8 segments, while paramyxo is unsegmented. Okay. What's the difference between the hemagglutinin and the neuraminidase glycogen spikes uh, in paramyxo and orthomyxo? Orthomyxo has 2 separate spikes, while for paramyxo, they are combined in one spike. All right? Hence the term paramyxo. Orthomyxo, segmented, and then there's 2 different spikes. Paramyxo, they're all combined in one glycogen spikes. Hence, it's para. All right? Para means all over. All right. This is the protein present in paramyxoviruses that causes the cells to clump together. This is what you call the fusion protein, or the P protein. Please remember these things because this is important for us. Okay. Multinucleated infected cells in paramyxoviruses viruses are known as what you call a syncytia. All right, these cells have a similar appearance to that of herpes viruses and herpes viridae infected virus and retroviridae infected viruses. So multinucleated cells infected by Pyromix of Viruses. Okay, they are known as a syncytia, and then it's similar to those of the Retroviridae family and the Herpiviridae family. And what are the other ones? I ask this question again. Um, I'm gonna ask this question again, and hopefully the people in the chat are active. What are the members or what are the viruses under the Paramyxoviridae family? You already have one, RSV. What's the other one? Anybody?
Sir,
Oh, so yes.
is measles one of them po? Measles po
Uh-huh.
ang sa akin sagot po.
Yes. Uh-huh.
And also mumps.
Uh-huh. What else? Yes, people are active in the chat. I love it. Because they think
Para-influenza po.
there might be a prize. Hence, the people are active. Para-influenza. Okay. Very good. Very good. Very good. Very good. All right. Wonderful. 'Di ba? Napaka-active nila talaga. Kapag ka talaga merong motivation, I think the students are much more active. All right. We'll try our best to give out prizes throughout our lectures so that you guys will be motivated. 'Di ba, Myra? Mayra? No? Mayra? No?
Yes, sir.
Are you are you uh are you expecting to get a prize sometime today? Hopefully.
Saks lang, sir.
All right. Early Christmas gift for some of my students. All right. Now let's go on to the somebody said something in the chat. I hope it's academically related. What's Hendra? Ewan ko sa'yo, Jonah. Sumasakit. Joanna, ha? Ewan ko sa'yo, Joanna. Baka wrong send lang yan. Napunta lang dito sa group chat, ha? Anyway, let's move on to what part of the respiratory tract in adults and in children are infected by RSV and parainfluenza. Well, for adults, it's actually just the upper respiratory tract, and for children, it's the lower respiratory tract. That's why physicians are much more, especially pediatricians, are um, much more careful to respiratory infection, are much more mindful of respiratory infections in children because to it can lead to pneumonia or viral pneumonia for children. Okay, so that's why that's the reason why I added this one. Okay, now what is the term used for a para-influenza infection in the larynx and the upper respiratory tract? It's also known as croup. Please don't forget it. Okay, so when you hear the word croup, it's para-influenza. Okay, it's associated with para-influenza. Now, RSV, you already transcribed it earlier for me, so I think it's gonna be redundant if I ask it again. But basically, it's respiratory syncytial virus. What is the difference between RSV? The structural difference between RSV and other viruses, the glycoprotein spikes. It lacks a glycoprotein spike. Okay, that's the only difference between the other ones. There's no glycoprotein spike in your RSV. Okay, so mumps, measles, and parainfluenza, they have glycoprotein spikes. But in the case of RSV, there's none. Now again for a cash prize. Oy. I will give 15 seconds
Sir,
on the
ready na
clock.
po.
All right, naka-ready na sila, oh. Regina, please don't forget to email me your email. To email me. You're no longer eligible for this cash prize because we don't want to see. We don't want to. We don't want to have. We don't have. We don't want to have be associated with any corruption. So only once lang ang students pwedeng magano.
Okay po,
During a
sir.
lecture. During a lecture, you can only win once. But on another lecture, you may be able to win once again. 50 pesos on the line, guys. And I will put
Okay,
15
sir, ready?
and I will put 15 seconds on the clock. What is the difference again between the glycoprotein spikes of Ortho Mixoviruses and Pyromix of Viruses. 15 seconds. Starts now. Wala sumasagot. What's the difference between Ortho Mixoviruses and Pyromix of Viruses in terms of glycoprotein spikes? 2, 1. Oh, nobody was able to answer. All right. We'll give an extra 5 seconds. Orthomixo separate spikes, paramixo combined spikes. Wonderful, Noreen. Wonderful. Taray talaga, active talaga Sir Marco mga students kapag ka may cash prize. Alright, wonderful, wonderful, wonderful. Wonderful. So 50 pesos for 50 pesos via GCash. Napaka-active ng mga students talaga. I love it. Pagka merong motivation. Alright. Now, how many hours do I still have, Adrian?
Sir, 1 hour and
1 hour
3
pa.
minutes po.
My gosh, wala pa sa kalahati itong Virology
Sir,
pa
kung
rin.
sa sir, sabi niyo naman din kasi mag-extend kayo. So, pwede naman hanggang 6?
Okay.
I mean, if it's okay with you.
It's okay with me. I have nothing else to do here.
Ako din naman, actually.
I have nothing else because I want to take the extra time that I was taking a break last week because I was working in the hospital. Now my full focus is on you guys.
I mean, okay lang naman sa inyo class, no?
Yes, sir.
Ayan, Joanna Batar says more prizes.
Yes, sir. Okay
Grabe,
lang po.
may kapalit pala yung OT nyo. Lugi
More
pa kayo sa
price,
OT.
sir.
Yeah. So yeah, it's okay with it's okay with me. I have nothing else to do after this. It's an extra hour.
Maagang pa, boss ko. Merry Christmas!
Yes. All right. Thank you. Now, Noreen, I want you to email me your number. Hopefully it's a globe number because my mom has my mom has difficulty sending money through other networks. Okay, so we only send numbers via globe Gcash. All right. Now let's move on to okay po. All right. Email please. And please put a smiley face on it. It would really make my day. What organs are usually involved in mumps? Parotid gland and the testes. Okay? So the parotid gland causes parotitis, testes, or chitis. What is the term given? As a set of signs, symptoms that arise from the presence of rashes and measles. It's a difficult question. I'll try to remember. Something to do with prodrome. Let's try to see the next slide. Yeah, measles prodrome. Okay? And prior to the appearance of a rash in measles, what term is used for the red base and the whitish blue lesions in the oral mucosa? This one is something that you must know. Anybody in the chat? The rash that appears as a red base with a white to bluish lesion in the oral mucosa.
Sir, public spots po.
Okay, thank you. But again, this is a question that's not prized. So thank you for answering, even though there's no prize. Complex spots. Usually my questions, guys, questions that have prizes are the ones that have been asked maybe 50 or 100 slides ago. Okay, so if I ask questions, if you participate in these types of questions, it would be wonderful. But I will ask this probably in slide 1, slide 3, so that you guys would have more time to search for it. All right. So describe the spread of a measles rash. So again, unlike chickenpox, it doesn't start from the periphery. It actually starts from the face, and then it appears on a downward spiral, and then it disappears. And then what happens is that it starts from the face, and then it disappears downward as well. All right. So facial rash that spreads down. Okay, and then rash from the face disappears, and then it goes down again. All right. So that's how measles usually a measles rash appears. Now let's move on with Pyromix of Viruses. Different virulence factors. I think this particular table that I will show you guys is something that might be useful for you. All right. So all of them have F protein. All of them have F protein. But it is only para-influenza and mumps that have heme agglutinin and neuraminidase in their glycoprotein spikes, whilst measles have heme agglutinin only. And as I mentioned before, RSV is the only one that doesn't have the glycoprotein spikes. All right, good, wonderful. We can move on. We'll move on to Papovaviridae. Oh, Papovaviridae. What are the family of What are the viruses under the Papovaviridae family? Please give me an answer. Somebody from the chat, whether it's chat or through the microphone, it will be fine with me.
Sir?
Oh, baka active ni
Uh,
Daniel ngayon. Sige, sige.
Hindi po
Sige,
pangangailangan.
push.
Polyoma virus po and
Polyoma virus.
Aha.
inoculating virus.
All right. So, okay. Wonderful. Medyo hindi ko naintindihan, but yeah, sure. Thank you for participating. But you mentioned some of the correct answers, so I will not take that away from you. Yeah. Papilloma virus, polyoma, and you forgot simian vacuolating virus. This is actually something that you have to remember as well. All right. And yeah, before we continue on, I would like to ask Adrian if it's still working, if our live stream is still working.
Yes, sir.
All right, wonderful.
It's still working po.
Okay, please remind me if it's disconnected. Thank you.
Okay po. Thank you, sir.
All right. Which virus under the Papovaviridae family is associated with warts? Oh, this one, genital warts, another form of genital warts. But this one, the warts are larger. This is papilloma virus. Okay, papilloma virus. And there are two members of viruses under the polyoma virus. So there's papilloma, and then we have polyoma and SVV or the simian vacuolating virus. So please don't forget these three viruses under the Papovaviridae family. All right. So how do you remember the viruses under Papovaviridae viruses? The papovs. Papovs. So papovs. What's the S? Simian simulating vacuoles. Okay, that is another mnemonic for you guys to remember. I'm I wasn't able to give you guys mnemonics for the other ones, no? But anyway, let's move on. Papovs. So it's pretty easy for you to remember. Papova. Polyoma. S is SVV. Right? Easy? Easy peasy. All right. Which viruses under the Papovaviridae family is associated with warts? As I mentioned before, it's the polyoma virus. And then we have two members under that one. We have the BK virus and the JC virus. Uh-huh. Now, the BK virus and the JC virus. Basically, they are just call this um, members under the polyoma virus family, under the papovavirus. They're just two strains, the JC virus and the BK virus. I was supposed to give you guys a transcription of the BK and the JC, but unfortunately I lost it. Don't worry about it. It's not a question in the board exam or any of the certification exam. So it will not be, I don't think it will be important. It's pretty uncommon for them to ask it for you guys. Anyway, just remember, I think the takeaway from this particular lecture is that Papova, polyoma, and simian viruses are under the Papovaviridae family, and then yeah, they're associated. One of them is associated with warts. Uh-huh. And let's try to talk about the SVV. Oh, from the name itself, what is the host for SVV?
Monkeys.
Monkeys. Yes, from the name itself, simian. Right, wonderful. Thank you. Nag-aalala sila. Nag-uunahan na, parang feeling ko naging mas active kayo. Like, the attentiveness became came from 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 a 1 to 100 in a matter of seconds when I started giving out prizes. So I love it.
Sir,
I love the
sp-
attentiveness. Aurelian,
spirit of spirit of Christmas po.
yes. 'Di ba? So
Kaya po kami active.
pwede kayo magsabi na namamas ko po. Yeah, I think that would be our contest rules. If you're going to answer some of the questions that are priced, sir, namamas ko po,
Ninong
my answer is. Ganon dapat ang answer ha. Is that okay with
and
the
I,
class?
thank you.
Is that okay with the class? Does everybody agree?
Yes, sir.
Si Rainiel lang nag-agree. Para si Rainiel lang.
Sige po, sir.
All right, so let's practice kunwari. Let's practice. All right, let's practice this once, okay? This is not priced, ah. What is the host? What is the host for your simian vaculating virus? Adrian.
Sir, namamas ko po, sir. The answer is monkeys po.
Okay.
Simian
So that's
monkeys.
how you answer, guys, huh? Next time I ask a question about um about that's priced. That has a price. All right? Okay, so what cells or tissues does papilloma have affinity with? Is actually anything or any cell that has epithelial cells or keratin or any cell that is keratinized. So again, the reason why I said it to you guys is because these are the cells that have these cells are responsible for, or these viruses rather are responsible for warts. Okay, HPV. All right, HPV. How many? It's not a price question. How many strains do we have? Anybody in the chat, whether advanced or Google search, doesn't matter. Let's try. Um, who else is who can answer this
Sir,
one? Oh,
Sir,
100.
oh, yan na. Oh, Regine, sige. Regine, ikaw
namamas
naman.
ko po, sir.
Regine, ikaw naman. Walang price naman, Regine.
200, sir.
Walang price, Regina. Huwag ka masyadong abuso. Regina, walang price yon. Mamaya-maya pa ako. Wala pa ako sa mood magtanong ng may price. Wala pa ako sa mood. Pag nasa mood na ako. All right. Indicate the HPV strain associated with the following: plantar warts, common hand warts, cervical carcinoma, and genital warts. We have HPV1 for plantar warts. And I will show you guys a picture. I actually put a smiley face on it during our 10-minute break earlier. I actually put a smiley face on it. This is the appearance of the warts in the genital. Yeah. So common hand warts HPV 2 and 4, and then cervical carcinoma. I put it on bold letters for you guys to remember. Why? Because 16 and 18 is the only one associated with genital warts. Okay? Or genital carcinoma. Okay? Hence the smiley face. Please remember this one. I think if I put pictures on it, people will remember it faster. All right. Herpes virus, that is, the cervical carcinoma can be screened with what test? It's a histopathology recall question. Um, it was asked in my friend's board exam 2020, 2020, 2014, I think. 2014. It's a histopathology question or histopathology recall question. Cervical carcinoma is screened with what test? Anybody? What about Charisse? Mera. Okay,
Pap
Mera.
smear.
Huh?
Pap smear.
All right.
Pap smear,
Wonderful.
sir.
Wonderful. Pap smear. Thank you. Di ba, di ba very ano na sila ngayon, attentive, tapos wala nang question mark, wala na yung paps? Pap smear, sir. Wala nang ganon. I love it. I really love it. I really love motivating students to study. But I love it more when they're motivated because of cash. I love. All right. Let's move on. Um, wait. Where's my presentation clicker? All right. So pap smear. This is how you collect a pap smear. Okay. Now, this is the site of latency for polio virus. This is that places the kidneys, of course, and reactivation of the BK and the JC virus is actually different. Okay, one one thing that you need to remember is that they occur in on patients who are immunocompromised. They might be infected before, but then no symptoms again because of latent because there's latent infection. But when it activates, the BK virus causes hemorrhagic cystitis. When I say cystitis, where does it occur? In the bladder. All right? And then the other one is multiple focal lymphadenopathy for JC or PML. Sorry, I have to correct myself. Progressive multifocal lymphadenopathy. All right? So BK virus and the hemorrhagic cystitis is associated with hemorrhagic cystitis or viral hemorrhagic cystitis. And then JC will then be JC virus is associated with progressive multifocal lymphoencephalopathy. I know it's a long, it's a mouthful, but you guys have to at least try. Now, this is a question for you guys. It's important. Why? Because you guys are medical technologists. All right. You will be your job will be in your future jobs will be working in the laboratory, and it's important for you guys to know the samples that you're going to use for BK virus because it causes hemorrhagic cystitis or infection in the bladder. You need to use the urine of the patient, and for encephalopathy, which is caused by JC virus, you need to use either tissue sample from the brain or CSF. Sir, brain tissue talaga, sir. Yes, you have brain surgery, especially in other countries. Unfortunately, I think in the Philippines, I'm not sure if they're doing brain surgery in the Philippines or they send it out, they send the patients to nearby countries with better infrastructure. I remember there was a case of somebody who had to undergo some sort of open open uh, conscious brain surgery. So you guys have to remember that in the Philippines, I think the medical facilities are still not as advanced. So it's very rare for you guys to see brain tissue, but it's pretty common to see CSF samples in the Philippines. So yeah, right. Now let's move on to parvoviruses. Based on size, the parvovirus is considered as the smallest and the simplest virus. Okay. I think this was asked in the PAMET quiz show way back when I was still a third year student who just attended and just wanted to see it. The question was presented in, wait, let me check my notes if it's, yeah, it was asked in the PAMET quiz show. It's the smallest virus and the simplest virus. These are your choices: Picorna, Parvo, Pox, and Papilloma. All the choices sounds all the choices sound are in C, and I think the goal of the question was to confuse the students to answer. So yeah, Parvoviridae is the answer to the smallest and the simplest virus that we will see. All right. And now, what disease is caused by Parvoviridae? It causes erythema infectiosum. So again, I need you guys to put a star on this particular question because it is important, especially in microbiology questions. Which of the following viruses causes the fifth disease or erythema infectiosum? Or patient came into the emergency room with having fever and a sim, cheeks resembling slapped appearance or erythema in the cheeks. So that's known as erythema infectiosum, slap cheek appearance. All right. Anemic crisis caused by Parvoviridae is what you call transient anemia, aplastic anemia. Okay, these are viruses that can actually cause aplastic anemia. Yes, viruses can cause aplastic anemia as well. And if you're going to study hematology, you will encounter this one again. I'm pretty sure of that one. Aplastic anemia. All right. This is known as a human parvovirus. This is the only serotype that you will remember. Don't worry about it. Only parvovirus that you need to remember is B19. All right, parvovirus B19. And now we're going to move on with picornaviruses. Okay, so the origin of the name for picornaviruses is the piccolo. Piccolo. Okay, why do they call it a piccolo? Because it's small. It's Italian for small. It was discovered by an Italian virologist. I forgot his name. I didn't write it, but it was discovered by an Italian virologist. He named it as a piccolo because it's a small virus. Based on size, it is the smallest RNA virus. All right, not to be confused um, with your parvovirus. Huh? So picornavirus is the smallest RNA virus. The smallest overall virus is the parvovirus. All right. So the two divisions of parvoviruses, we have rhinoviruses and the enteroviruses. All right. Can you guys still remember what are the enteroviruses? We have your Coxsackie A and Coxsackie B. What else? Enteroviruses. There are two. Coxsackie A, Coxsackie B. What else? Enteroviruses.
Echo virus po.
Ha?
Echo virus.
Oh, nakalimutan na nila kanina. Kanina tinanong ko, kanina nandun yun sa slide. Actually, it's somewhere around slide number 15 or 16. Ah, no. Slide 70 pala, 70. Somewhere around those, somewhere around the lines of those one. Enteroviruses, there's three. I will leave it to you guys to answer later because we have to move on. All right. Um, they infect what cells in the body? They infect the intestinal epithelial cells as well as lymphoid cells. And there are 5 subgroups of enteroviruses. I've mentioned it before. Echovirus, Coxsackie A, Coxsackie B, hepatitis A, and the new enteroviruses or the neo-enteroviruses. What is the mode of transmission of enteroviruses? What is the mode of transmission? Without looking at the next slide, what do you think is the answer? Mode of transmission for enteroviruses. Ano guys? Enteroviruses are transmitted via what? The mode of transmission.
Fecal oral po.
Wonderful, thank you. Who was that? I didn't see the name. Who was that? Charisse. Ah, okay, Charisse. I was waiting for you. I intentionally didn't put a slide, an answer to that slide, because this is actually another prized question. Unfortunately, what was our rule, Adrian?
Namamas ko po.
Yeah. Charisse. Sayang. Charisse. Nakalimutan ni Charisse yung rule. Now, let's move on to polio viruses. The specific cells infected by a polio virus is your Peyer's patches and motor nerves. I'm sorry, Charisse, but you were not able to get the prize for today because you forgot to follow the rules. Remember when you're in the board, this also serves as training for the board exam and even in your examinations. Erasures and everything are supposed to be avoided in the board exam, huh? And all of the following, nataranta din po ako sumagot. Okay lang, Charisse. You still have a lot of time to answer some of the the following questions. Describe the infection based on age. Children are less likely to develop poliomyelitis. whilst older individuals are more likely to get poliomyelitis. All right. So there's a high probability for adults. So be careful. But everybody I think has their own I think everybody has polio vaccines. I think I this is the only disease or this is the only viral infection that has been eradicated from the world. Yeah. And there are three stages of this virus or three disease manifestation. Mild illness, usually fever or common signs and symptoms of a viral infection. And then you have what you call aseptic meningitis, within which there's no bacterial growth, hence the term aseptic meningitis in the cultures. And then paralytic poliomyelitis is the one. Usually you will see this in polio ads because we have Polio Day in many countries. We have World Polio Awareness Day. So you will see them as paralytics. Now, how is Coxsackie A and Coxsackie B differentiated from each other? Mice, the effects on mice. Why? Because this was actually discovered on mice. Now, differentiate the symptoms caused by Coxsackie A and Coxsackie B. Um, I don't think this will be important, but just for you guys to remember, Coxsackie A causes herpangina, whilst Coxsackie B causes pleurodynia, myocarditis, and pericarditis. I would put it on the uncommon bin or the nice to know bin in several parts of your brains. All right. Now, what is the disease caused by rhinovirus? Adrian,
Yes, sir. Yes
what
po.
what is the disease caused by rhinoviruses?
Common colds po.
Wonderful, thank you. I was waiting for namamas ko po. Oh, nasad ako. Nasad ako. I'm
Sorry,
waiting
sir.
for namamas ko po.
Sir, so ibig sabihin po ba yung ruling po natin kahit hindi po price question basta magsabi lang po ng namamas?
Um, I will say it like this para wala tayong confusion. Um, okay? When I say this is a prized question, please bear in mind that you have to say namamas ko po. And then sometimes I will ask surprise prized questions like the ones that I asked like the ones that I ask on like the one that I asked Charis, right? Said I was waiting for namamas ko po. Yeah. So overall, I think
Sorry, sir.
I think the overall rule would be always answer like that. Would it be better? Right?
Sige.
Everybody, si Adrian lang ang nag-agree.
Yes po sir, agreed po.
Ayan,
Yes.
ayan na nag-agree na lahat sila. I love
Yes
it.
po.
Bigla silang naging attentive, Norin, no? Norin, naging attentive sila, no? I love it. All right, so yeah. Temperature that is ideal for cell cultures of rhinoviruses is around 30 degrees Celsius. Why is it important? Because sometimes you might be working in a research laboratory and rhinoviruses are pretty much the only thing for fresh graduates to work on. Yeah, because it just causes common colds. So I think it's important for you guys to know this. And enterovirus serotype for HAV, enterovirus 72. All right, hepatitis V. Hepatitis A is enterovirus serotype 72. All right. Now let's move on to pox viruses. There are three diseases associated with pox viruses. Please don't forget these three. ORF disease, or the ORF, molluscum contagiosum, and smallpox. All right. Based on the structure, pox viruses is the largest DNA virus, and it's the most structurally complex. What is the shape of a pox virus? If you remember from last week's lecture, this is a prized question.
Sir,
Oh,
naman po po, brick shape
Po.
box.
one by one muna. Raise your hand. Mayra. Mayra was the first one.
Namamas ko po, the answer is shape or back shape.
Ah, wonderful. I love it. I love it. So please send me your email later. So who are the people who will get the prize? Um, who are the people here? Noreen. I think Noreen is one. Regine and Mayra. Noreen, Regine. All right, somebody's keeping track. Thank you. Thank you. Thank you, Adrian. Thank you for keeping track. I love it. All right. Next. Next. Structurally complex. Now let's move on to describe the shape of a pox virus in the genome. It's box shape or brick shape. All right. So the genome is like this. The genome is dumbbell shaped. All right. Trivia. The last case of smallpox was seen in the year 1977. It's just trivia. Might not need it, but I just posted it there. However, since I created this lecture a couple of years back, I think it was a decade before, I think it was 10 years ago when I first created this slideshow. There was one case in 2018 where somebody got smallpox, and I would like to omit that, but there's still not a lot of credible information on this on literatures. So I would still stick with 1977. All right, because there's none of the books that I've read about virology have any changes. It's still 1977. All right, where was the vaccine from smallpox derived? It was derived from the Vaccinia virus, which is called cowpox. All right. What are the reasons why smallpox vaccination eliminated the disease? Vaccination created solid immunity and long-lasting immunity. There is no animal reservoir for smallpox, and clinical presentation of smallpox is obvious. Yeah, so that's the reason why it was eliminated or eradicated worldwide. Smallpox is actually very deadly in before the time of Edward Jenner. So we have so much to be thankful for because Edward Jenner used variolation on cows that are infected with cowpox. Now, how is smallpox transmitted? Again, this is because the reason why it's deadly and it's because it's avoidable it should be avoidable before way back in the 1970 before 1977 is because of the fact that these things are transmitted by respiratory droplets, especially for smallpox, and molluscum contagiosum and ORF are transmitted by direct contact. Okay, so smallpox is inhalation. Is transmitted through inhalation rather. Clinical presentation of molluscum contagiosum, the contagiosum and ORF are both presented by localized dermatologic conditions, while smallpox is more of a whole body experience. I would say it's more of a whole body experience. These two, they're just localized dermatologic conditions. All right, describe the appearance of a lesion consistent with molluscum contagiosum, a large bump with a central dimple. This is your mnemonic, guys. You scum, you ugly mole. See, molluscum and ugly mole. See, it's a large bump with a central dimple. All right, Rio viruses. We're gonna move on with Rio viruses. I'll give you the origin for the name. It's actually an acronym. It's actually respiratory enteric orphan. Oh, it's actually the name for the virus originates from an abbreviation, respiratory enteric orphan. What orphan? What do you mean by orphan? We'll talk about it later. Now, what virus under the family Viridae is associated with human infection? This is the rotavirus. Guys, please don't forget about rotavirus because it is the number one cause of infantile diarrhea? It was a board exam question, so please, please, please put a large star on this or highlight this or pin this or do whatever with this because this one is a board exam question from a test bank. Okay? So number one, acute infection or what you call the number one cause of infantile diarrhea, right? Rotavirus. Next, bacterial disorder that's considered to be much more than rotavirus in terms of dehydration, cholera. So they're almost at the same level, but much more worse, of course, because they present the same they present the same clinical picture. However, rotavirus is much more I would say much more innocuous. All right. Now, let's move on to retroviruses. I asked before, what are the two medically important retroviruses? Um, can you please help me out here, Noreen, since you're getting, no, Myra, sorry, since you recently got, since you recently won a prize earlier. Myra, can you please help me out?
Yes, sir.
What are the two medically important retroviruses?
Human T-cell lymphotropic virus
Uh-huh.
and HIV, human
Wonderful.
immunodeficiency
Thank you so
virus.
much. And how do you feel right now that you won 50 pesos GCash? Charing in interview. How do you feel right now? Before we
Mababayaran ko na yung utang ko sa Globe.
wonder, o di ba nakakatulong na. O di ba? Nakakatulong na. Nakakatulong pala ang ating ano. Marami pala tayong natutulungang mga kababayan natin. I love
Kuya
it.
Will,
I love it. Marami tayong natutulungan kababayan natin dahil dito sa ating lecture na to. All right? Thank you so much, Mayra.
Thank you din, sir.
All right, human T cell lymphotrophic virus. So human immunodeficiency virus. Now let's talk about HTLV first before we move on to HIV because that will be, I think, a bulk of the questions in the board exam. So HTLV was first discovered in the is commonly distributed in the Caribbean, and HTLV2 is commonly distributed in the Korean and Japanese peninsula. Peninsula, I would say peninsula. Yeah. The islands of Japan and Korea na lang para mas cute. All right. So what are the diseases caused by HLV1, HTLV1, HTLV2, tropic spastic parapheresis, and sorry, HTLV2 has no known disease process. It just causes the inactivity of your T lymphocytes, or rather a decrease in the number of T lymphocytes. Now, trivia. What form of leukemia did the discovery of HTLV2 begin? It's actually a variant form of hairy cell leukemia. Okay? And you will see this most of the time when you're studying about leukemias. HTLV1 and 2 are usually associated with leukemias. All right, especially hairy cell leukemia. So if it's not going to be asked here in microbiology or virology, it will be asked in your hematology classes. Now, let's talk about the causative agent of acquired immunodeficiency syndrome, which is HIV. Um, the reasons why a retrovirus was suspected when the pathogenesis of AIDS was not yet understood was because of the following. The infection was transferred via blood and blood products, delayed onset of signs and symptoms, immunodeficiency can occur in other animals as well, and destruction of T cells are seen with HTLV. That's the reason why they suspected a retrovirus because they have the similar signs and symptoms of HTLV. They were transferred. The disease was transferred from blood and blood products, and immunodeficiency occurs in humans. Actually, we have what you call SIV virus, which is another retrovirus that is known as the simian immunodeficiency virus. We also have the CIV, canine immunodeficiency virus, which causes which causes similar disease progressions to that of HIV. Now, we'll talk about T cell cultures as stimulated by interleukin 2. Um, um, what would happen is that the virus shares a close homology with HIV 2. There are actually two types of HIV, HIV 1 and HIV 2. So please be mindful of those ones. And usually machines right now are sophisticated enough to have reagents and um, chemiluminescent probes to detect both HIV-1 and HIV-2 directly without no, not the antibodies, rather directly without having to get a different aliquot. I've I was fortunate enough to be trained in serology for I think almost a year, and I was running HIV screening tests. Of course, these are screening tests because it requires a much more advanced training, especially here in the Middle East, to run confirmatory tests for HIV. So in this case, you have two reagents. You have only one reagent that can detect both HIV-1 antibodies and HIV-2 antibodies, and usually they come in a package right now. And this is not a commercial for Abbott systems, but Abbott is probably one of the leading companies who whom created chemiluminescence, or rather promoted chemiluminescence for the detection of these two antibodies. All right. Now, SIV it shares the same homology as HIV-2. Geographic distribution of HIV-1 and HIV-2. I think this is the reason why I put it here, and the reason why it's important in this discussion, in our virology discussion, is because of the fact that many people are still confused with within which HIV has been isolated in, and HIV is HIV-1 is the most common one. It's located almost everywhere around the world, whilst HIV-2 is common in Western Africa. So, because a lot of Western African countries are economically challenged, the the spread of HIV-2 is somewhat limited. So HIV-1 is more because there's a lot of people traveling around these parts of the world, Europe, America, and Asia. Um, that's the reason why that's the reason why I think many scientists believe that HIV-1 is the number one cause of the HIV pandemic. So yeah. All right. Now, HIV distribution of HIV-1 and HIV-2 is actually quite interesting because HIV-1 is more common in males, and HIV-2 is equally distributed among males and females. Now, is this an important statistical analysis? Yes, it was a question in the ASCP, in an ASCP question bank recall question review. Okay, so please try to be mindful of these things. I think the easiest way for you guys to remember these data, or to at least have an idea or a gist of the data that I'm presenting, is just remember HIV-1 is spread worldwide, while HIV-2 is um, somewhat isolated. I didn't say isolated only in Western Africa. It's isolated somewhat to a certain degree. It's isolated in Western Africa. And then when you go to gender distribution, it's more on males and females. It's more on males rather than in females for HIV-1 because, of course, the people travel, and it has been associated historically as a gay-related rather, or homosexual relations, or um, man-to-man sexual relations. And of course, I think the previous terminology of this was I don't want to sound controversial, but I will give you guys a historical preview of HIV. It was considered in the 1970s as a gay immunodeficiency syndrome. It wasn't known as human deficiency syndrome. It was known as uh, but then activists in specifically in America, um, they they discovered that it wasn't just spread among males who have anal intercourse all the time. It was also spread by blood products, and it's not appropriate to call it it's not appropriate to call it as gay immunodeficiency syndrome. So they changed it, of course, but there is the stigma that, of course, when you get infected by HIV, you're immediately associated with a person who you're immediately associated as a person or stigmatized as a person as having same-sex intercourse, specifically on the male side. All right, so this is actually not just a lecture on how you answer the board exam, but rather to get you a little bit more educated on how HIV spreads. All right, so hopefully you guys would still remember me when you guys go to the board exam and said, ah, yeah, I remember Sir Manuel told me about this. Because this was a question in this was a question in the question bank of ASCP, the BOC, Board of Certification of the American Board of Certification. All right. So what is the nucleic acid content of the retroviral core? Well, it basically has it's simple. It has one dimer of the single-stranded RNA. So it's just a dimer of ssRNA, or what you call single strand of RNA. Now, what enzymes are found in the retroviral core? This is important not only in your microbiology class, but also in serology, as well as in if you're going to take advanced courses in molecular biology, because the enzymes that are found is what you call the vital proteases. Vital proteases prevents the virus from becoming from disintegrating. All right, because sometimes they might go into the body and there are certain chemicals or chemokines that can destroy the cell, that can destroy the viral particle, and viral protease is important for that one, as well as allowing the core of the virus to penetrate the cell membrane. The next one is the reverse transcriptase. We've already mentioned what it does. It it transcribes it transcribes the genetic material of the virus from RNA to DNA, thus allowing it to be implanted by the third enzyme, which is your integrase enzyme. What is the importance of integrase? It allows the genetic material to be integrated into the host cell. So the viral genetic material will then be integrated into the host cell. Specifically, what cells are the target of your HIV infectious HIV virions? Please, anybody in the chat? Anybody? Nobody? Hello? CD4. Ay, bumalik na naman tayo, Joanna, sa ano, no? Sa not sure. CD4. Charot. CD4. Sir, bumalik na naman tayo sa ano. Sir, can you please repeat the question? Well, my question was, what is the target cell of the HIV virion? T helper cells. All right? And what is the CD molecule present in? Yeah, that's correct. Yeah, that's correct. Joanna, bakit ayaw mo na? Joanna, your answer was correct. It was just a little. But unfortunately, Joanna, it's not a prized question. So there's no prize, unfortunately. So we have to move on, Joanna. All right? It's okay po. At least we were able to learn, right? Correct? You were able to at least absorb something. Sa knowledge tayo, sir. Ah, yes. Yan ang gusto ko actually. I actually love the students who participate not because of the monetary benefits, but rather the knowledge-based benefits of subject matter. Now, the main capsid of the protein of the retrovirus is actually what we call the P protein or the Pol protein, P24. It is the early marker for HIV infections as well, P24 or the Pol protein. And then we have what you call the surrounding matrix protein. This is the glycoprotein matrix. What is the function of the protein matrix surrounding the retroviral envelope? It holds or anchors the glycoprotein spikes of the virus. Again, we're gonna go back to the realms of glycoprotein again. But there are two specific glycoproteins that you need to remember. This is important. Please put a mark on this one because this will be asked in an exam. What are these type? What are the glycoprotein spikes that you need to remember? 122 and 41. GP42, GP41, and GP100, GP100, GP120. All right. Now, let's move on to the viral genome. Transcribe LTRS. LTRS stands for long-terminal repeating sequences. The function of long-terminal repeating sequences is as follows. They are sticky ends, the portion of the gene that is recognized by your integrase enzyme. And they are the promoter or the enhancer, not enhancier, enhancer portion of the gene that is coded to promote viral transcription. All right. So long-term terminal sequences. So that is the genetic sequence that is usually recognized by the integrase. It's actually known as the sticky portion or the sticky end. What part of the HIV genome? This is the part of the HIV genome that codes for the protein inside the viral envelope. This is known as what you call the Gag genes. Or Gag stands for group antigen. Group antigen. Gag gene. All right. So what are they? They are the proteins in the viral envelope. All right. Next, what are the proteins encoded by the Gag genes? You're going to you're going to see this in the viral envelope. It is the nucleocapsid, the capsid, and the matrix protein. And this is the HIV gene that codes for the enzymes inside the viral capsid, Pol gene, okay, and then we have the enzyme gene. What are the enzymes? This is the HIV gene. We're talking about the gene first. So the Pol gene is responsible. Pol, all right. It's not Paul as in Paul, Paul, Saint Paul. No, it's Pol gene, Pol. Okay, and the enzymes that are coded are your vital proteases, integrase, and the reverse transcriptase. So again, um, vital integrases are important because they are the ones that cleave the Gag and Pol genes, and they provide the virus with precursor genes. All right, and then the function of vital protein. Okay, yeah, I've already mentioned that earlier. What is the HIV gene? What this is the HIV genes that codes for the GP genes. We have what you call the ENV genes, short for envelope. All right, it's short for envelope. HIV gene that codes for the GP proteins or the glycoproteins is your ENV gene. What are the envelope proteins that we need to remember? You need to remember that you have 120 and 41. 120 is the head and GP41 is the stalk. All right. And this is actually important because when you do Western blot, these are the things that is required to be detected. Any positive line in any of the bands for GP120 and GP41 will indicate that you have a confirmed HIV infection. All right, glycoprotein molecules in the viral envelope are collectively known as what GP number? GP160. How do I know it's GP160? Inad ko lang -1 so I can remember, right? GP160. So the whole thing here, that whole thing over there, that one that looks like a thumbtack, it's GP160. Saan napunta sir yung 120? Yung 1? Actually don't know. But that is what is the lesson here is that it's based on literature, and I want you guys to know it because it will be asked in the board exam. All right. Main function of the glycoprotein spike in the viral envelope, it is the one that binds to CD4 receptors. It allows the CD4 receptors to become it allows the viral protein, the viral particle to be absorbed into the cells. All right, the regulator genes of HIV. Please don't forget the Rev, TAT, and NEF. Please don't forget these things because they will ask you guys about it. Which of the following genes are regulatory genes? What do you mean by regulatory genes? These are genes that are responsible for the replication of the viral genome so that go it doesn't become it doesn't become obsolete because because of constant transcription and translation of the genes, sometimes they become sometimes they become obsolete. Like imagine using the paper, a piece of paper, you write on it, you translate something on it, right? Keep on writing on it, and then there's no regulatory that can there's no regulation that can erase that can erase anything that you've written on it. And so what happens is the message encoded in your paper is either illegible or you can't read it anymore, which is the same thing that happens once once a genetic material has become overused. So these regulatory genes are important for HIV to survive longer in the present. So what is the protein product of the TAT gene? Well, it binds to the viral DNA and activates the transcription. See? See what happens? Function of the Reb protein, there's two. Splices HIV DNA to host cell to DNA host to the host cell DNA. Okay? It splices the host cell DNA to produce species of gag pole and ino or envelope genes. All right? So in short, it revs up the viral replication. Lastly, the functions of the ino protein is to provide or it binds the HIV splice DNA to increase viral production. All right. So those are your regulatory genes. Now, HIV gene that produces the antigenic products, the Gag gene. All right, we already know that. This is the part of the HIV virion that is hypervariable and antigenic, that has an antigenic viral region. This is GP120 and the reverse transcriptase. Would you believe it? The enzyme is actually a antigen. It's actually an antigen, the enzyme itself. All right, and this is subject to mutations as well. Mode of transmission for HIV, sexual intercourse or exchange of body fluids, and of course, parental route. Which gender is have high risk for contracting HIV? Would you believe it? It's actually the females. Yes, would you believe it? It's the females. Which gender is at high risk? Why? Um, um, Females are much more at risk because they get pregnant. They would require much more transfusion than males. Males only get transfusions during um, during during any sort of accidents. Females are also at risk for depression, and therefore they would more statistically they are more at risk of using illicit drugs. Okay, based on statistics, I'm not basing it on opinion. Huh? Females are also um, require drugs to maintain certain sorts of diseases. There's a lot of diseases for females that require parenteral introduction of drugs. Now, males, on the other hand, can have relatively can have relatively the same um, need for drugs. But the only difference for males is that they are most likely promiscuous. Hence the stigma of HIV infections. But I think nowadays, since I created this lecture in 2010, I think values have sort of changed or sort of flipped over into the male side. I would need to have some sort of a, so I think I should remove this one because this is on this is more relevant way back in 2010 when I was still studying for the board exam. Now if I think if I look at the statistics, it would probably be the polar opposite. So I'll try. Let's try not to focus on this one first. Okay. So HIV infection, cellular infection. Virions infect what cells of the body and why? As I mentioned before, they will affect. They will infect primarily T lymphocytes, monocytes, and nerve cells. Because the presence of CD4, which binds to GP160. All right. So rarely on nerve cells, but most probably helper T cells. 90% of the time they will infect uh they will infect they will infect T lymphocytes. But monocytes they will infect it primarily because they will infect macrophages primarily because if it's they would the macrophages will phagocytize helper T cells that have been infected with this one because they're not recognized by once the antigenic presentation has been altered by the virus, the monocyte will consider it as foreign. Nerve cells, very rarely we have a certain parts of the body that have CD4 markers. Okay, so it's very rare on that side. Now, let's move on to COFA. But in the exams, I want you guys to focus on a helper T cells. Okay, so the major target of the HIV virions would be helper T cells, but it can based on literature, it can affect it can infect monocytes and nerve cells depending on the state, depending on the transmission. All right. Now let's move on to the cofactor proteins that initiate the building process, the binding process of HIV. There are two ones. We have what you call the fusin protein or the fusin protein, which is responsible for the fusing of CD4 and CD4 GP160. And lastly, we have CKR5. I need to remember these two, of course, because HIV is a topic that is commonly questioned in the board exam. All right. Uh, which HIV binding protein is said to make a person immune to HIV? This is the one that they detect on patients who are immune to it. Is there immunity to HIV? True or false? Question in the chat for the chat box. Is there is there immunity in the in for HIV? Can a person be rendered immune for HIV? True or false? Write it on the chat, people. True or false nga? Yes. Ikaw talaga, ikaw talaga Adrian, ha? True or false?
Sir, namamas ko po, true.
Not yet. Binago ko na ba? All right. Who else? What about Joanna? Is it true for you or false? What about Mayra? What about Noreen? AJ said true. Okay. Mayra said true. All right. Joanna, trolls? Joanna, huh? Joanna. Yes, a person can be. Yes, a person can be rendered immune to uh to HIV. There are tons of research right now, especially now. I think 2018 was the year they reported 5 people to have been transplanted with bone marrow from irradiated bone marrow. Oh no, their bone marrow was irradiated and then they were transplanted bone marrow stem cells, and they were successfully they got successfully rid of the HIV infected T cells. All right. So what are the functions of the HIV enzymes in relation to cellular infections? As I mentioned before, again and again, vital proteases cleaves the viral RNA, okay, allowing the viral RNA to survive in the cytoplasm, of course, in the cytoplasm of the of the organism, or rather the nucleoplasm, sorry. Uh, Reverse transcriptase converts viral RNA to dimers of double-stranded DNA, and integrase splices the viral genes to the host cells. All right. Now, which process of viral replication of HIV contributes to the cellular killing of T lymphocytes? It is budding. Remember budding? The other one was what? Exocytosis. Budding, yeah. The other one is exocytosis. So budding is how because the HIV virus is an envelope virus, it will undergo by budding. And again, it's dangerous because it diminishes the cellular structure, or rather the cell membrane of the cell. Okay? Next, what are the stages of HIV infection? I think it's pretty straightforward. Acute viral illness, clinical latency, AIDS, and lastly, death, depending on the complication, of course. But most probably, the origin of the death, or the origin of the mortality is primarily associated with respiratory infection. There's a plethora of of infectious agents that can cause disease to a patient who has AIDS. So this is basically what happens. A summary rather of the clinical symptomology of the clinical picture of a patient with HIV who is untreated, who is left untreated. So acute viral illness. What's an acute viral illness? Clinical symptoms of common colds. Clinical latencies. The signs and symptoms are there, but lymphadenopathy is the signs and symptoms are not there, or not present, or not evident if you want to be more posh. Lymphadenopathy is unseen, or is evident. Okay. AIDS is depending on the immunosuppression. Signs and symptoms is dependent on the immunosuppression. How do we classify an HIV infected person with AIDS? How would we classify them? What tests do we need to do in the laboratory? Anybody? I don't think it's part of the I don't think it's part of the presentation. So I think this would be something that, yeah, it's not part of the presentation. I don't think it's part of the question. How would you test? I will give 10 seconds on the clock for the person to answer the question. Now you have to follow the instructions that I've given to you, because this is a prized question. There is a prize for this question. 10 seconds on the clock. You guys may begin. Raise your hand and unmute yourself. Nobody? 10 seconds. 10, 9. Nobody? What test can you use to classify someone? All right, somebody raised their hand. Who raised their hand? All right, Rainiel, try.
Sir, ano po ulit yung question just to be sure yung sagot ko? Kasi price question po to eh.
You're not following the.
Sir, I'm very scared to commit mistakes. It's a price question.
The question is, what test in the laboratory would you use to classify if the patient is HIV positive? If an HIV positive person has AIDS.
ELISA test po, sir.
Oh.
Ay, namamas po, sir, ELISA test.
Wrong.
The long
Regardless
version
of your
po
regardless whether you follow the question. I will give an extra 10 seconds for anybody to answer the question. Anybody for the steal? Nobody? All right, so that's the end. So what do we do? What do we do to identify to classify a patient with HIV? We do flow cytometry of CD4 cells. We do flow cytometry of CD4 cells, or we just do a CD4 count. You can say CD4 count. You can do CD4 flow cytometry. It doesn't matter. That would be I would have considered it correct. And another thing that you need to remember is if a patient who has been infected with uh, who's been infected with HIV to be classified as an AIDS patient needs to have less than 200 CD4 count, less than 200 CD4 count. So please remember it. It is a high yield question. It will be asked in the board exam most probably. All right. Okay. So what is the site of viral replication? Lymph nodes and macrophages. All right. Clinical latency of HIV infection is not considered as a true latency stage because of the slow rate of viral replication. All right, because of the slow rate of the viral replication, it's not actually a dormant stage. The virus is just replicating on a slower pace. All right. So yeah, and according to the World Health Organization, how much CD4 lymphocyte is needed to be considered as an AIDS patient? What was that answer? Anybody on the chat? Anybody on the chat? What was that answer? I mentioned it before. According to the World Health Organization, how much CD4 lymphocytes, CD4 positive lymphocytes, is needed to be considered as an AIDS case? 200? 200?
Sir, less than
AJ,
200?
sorry, unfortunately, AJ Aquino answered it correctly. Less than 200 cells per UL. All right, I know it's frustrating. Rainiel, I know it's frustrating, especially because of the new patch that is coming for Genshin Impact, but you have to be careful. Yes, but you have to
Sir.
be careful. I know.
Yes, sir.
Because of the current patch that is coming in less than 5 days now. I know. It's frustrating, but you have to focus. Less
Yes,
than
sir. I will.
200 cells
For
per
her.
UL. All right. So an infected T helper cell produces what glycoproteins in its cytoplasmic membrane? It produces GP160. All right, GP160, which is a combination of GP120 and 124. Vadding virions strip apart the cell membrane and the binding of the infected cell to CD4 cells. This causes the infected cell to be recognized as non-self, and then they will be phagocytized by macrophages, and henceforth they will infect these virally infected phagocytized cells would then infect the macrophages and then spread throughout the body. All right, that's how that's how it happens. What is formed when CD4 positive cells bind to HIV infected cells? They will produce multinucleated giant cells. Do we see this in the blood smear? No, very rarely. Okay, but I just put it there for your information, guys. Multinucleated giant cells are important in disseminating HIV virions through the body by the following. Through the body, through what mechanisms, of course? They allow the transfer of the virus from blood to out of the body they allow the transfer of the virus through the body without contact with blood. Okay? So yeah, and then it prevents the virus from neutralization by antibodies. So basically, they have what you call these multinucleated giant cells carry or ferry the cells to other parts of the body, and therefore they will then wreak havoc on different parts of the body itself. So imagine these multinucleated giant cells as jeepneys that ferry the virus throughout to different parts of the body. So yeah, papuntang Caloocan or papuntang where is capital nga pala? I think capital is near Fisher Mall if I'm not mistaken. It's been a very long time since I came back to the Philippines. Right? Capital is near Fisher Mall. Am I correct? Correct? Anybody?
Yes po.
Yes, it's still there. Near
Yeah.
what was that church? I can't remember what church it was.
Santo Domingo Church po,
Ah,
sir.
yeah. Yeah, wonderful, wonderful. I forgot about it.
Namamiss ko po, sir.
It's I'm terribly sorry. It's been a long time since I've taught in Capital on a personal on a face-to-face basis. So yeah, we still have how many minutes, Adrian?
Sir, until what time po ba tayo? 6 po.
Sabi ni Sir Marco pwede naman daw extend ng 1 hour.
Sige po.
All right, so
Okay po.
until 6. So how many minutes do we have? I don't have a timer in front of me.
So we have 49 minutes po.
All right, so I think we can finish the virology lecture and then I can skim some parts of some of the important parts of
Sige po.
bacteriology. But yeah, we'll continue with bacteriology tomorrow. We just need to finish this one because I think this is important for the board exam for you guys. All right, so what's the effect of HIV on B cells? They they activate polyclonal B cells. This forms constant mutation of the virus. All right, so polyclonal I wouldn't say polyclonal gammopathy. I would much rather prefer no polyclonal activation. I would much rather uh, call it a mutation rather than anything else. Um, which cell serves as a reservoir for hosts? As I mentioned before, these are your jeepneys, which is your what? Monocytes. They carry the HIV through the CNS. They can carry the HIV virus to or the human immunodeficiency virus to the CNS because of the fact that they can transfer or they can pass through the BBB. What is BBB, guys? Anybody? Anybody in the chat?
The brain barrier po.
That brain bar.
Nakakalimutan nila yung namamas ko po. I was supposed to give a prize for that one, but it's okay. We'll try again next time. Sayang. Nakalimutan nila yung namamas ko po. Please remember to follow the ano ha? To follow our sayang magbibigay sana ako ng prize for that one. What forms of malignancies can be associated with HIV? Because since we're talking about HIV transferring throughout the body, we also need to talk about the malignancies associated with AIDS. And this causes B-cell lymphoma and Kaposi sarcoma. Pero sir, kanina minention mo may isa pang virus na nagkakos ng Kaposi sarcoma. Who was that virus that causes Kaposi sarcoma aside from HIV? Anybody can answer?
HHV8 po.
H what?
HHV8.
What number?
8 po.
8. Okay. Unfortunately, again, you didn't say namamas ko po. So, you missed the opportunity. Now, what other viral DNA is present in patients with AIDS and HIV lymphoma? EBV DNA. Why is EBV DNA present? Why do you think ABV DNA is present? Because some patients have a latent infection. What do I say? But what do I mean? Why did I put emphasis earlier with latent infections? Because EBV DNA, some patients have already been infected with infectious mononucleosis, but they're not. The constitutions are their bodily constitution is sufficient enough that they don't suffer the effects of the illness. However, the virus is still there. It's just in a dormant state, and therefore, when HIV becomes present or predominant in the human body, it becomes active once again, specifically in a patient with B cell lymphoma and the person with AIDS. See the reason why I consider it why I put emphasis on latent infection in our earlier discussions. All right, this is the form of malignancy commonly seen in homosexual AIDS patients. Oh, commonly seen in homosexual AIDS patients. I think it's very controversial, but we still have to consider it. We still have to put it in under consideration because it's commonly seen. I looked at the statistics also. Personally, I looked at the statistics personally, and yes, it does happen on homosexual AIDS patients, particularly homosexual men, and that is Kaposi sarcoma, and it's associated also in patients with HHV8. Yes. Now, let's move on to the nitty-gritty. These are the things that usually are being detected. These are usually the things that usually that are 100% going to be asked in any of the exams that you might encounter in the future. The diagnostic tests for HIV. We have enzyme link immunoassays, or right now we can use chemiluminescence or chemiluminescence. Nagkekeme tayo. Alright? Kinikeme natin. Kasi baka mamaya, baka mamaya, kinikeme mo lang pala yung pasyente, false positive pala. And therefore, you need confirmatory test. O di ba? You have another mnemonic, guys. Another mnemonic. So please be mindful of those ones. So what are the the enzyme link immunoassay? And then we also have confirmatory test, which is Western blot. Now that we've finished with HIV, we can now move on to the rest of the viruses. Rhabdovirus has a characteristic shape, which is what? Anybody in the chat? Not a prize question, but I hope you will remember.
Bullet shape.
Wonderful, Adrian, and wonderful, Mayra. Thank you so much for participating. Unfortunately, that wasn't a question that has a prize, but thank you for participating anyway. Let's move on. The only rhabdovirus that is capable of causing human infections is rabies virus. All right, and rabies is transmitted by a bite from warm-blooded animals. Now, can you guys give me an example of the most common warm-blooded animal that can transmit rabies virus to an unsuspecting victim? Give me one. Give me one. Give me one warm-blooded animal. Dogs. All right.
Cat.
Can I get it from chickens? True or false? Can I get it from chickens? Adrian said false. Who wants to contest Adrian's claim and Mayra's claim? Can I get it from chickens? Chickens. No, no. False, false. Nobody would say true. Nobody would say true. Reina said false also. Adrian said false also. Joanna said false. Warm-blooded po sila. I don't know what you mean. Please unmute yourself and explain what you mean. Ems. Joanna. Joanna, sasampalin kita. Kung, kung nag-face-to-face classes lang tayo, Joanna, sasampalin kita. Ay, Joanna. Ems. Naloka ako, Ems. Wala akong magsasabi ng true. Kasi true yung tama. Theoretically, warm-blooded animals are basically anything in land. So even birds can carry the rabies virus. So most of the students answered no, but it is very rare. It is very rare. There have been cases in San Lazaro. Sir, tayo po may rabies din. Human po, I mean. Yes, you can get rabies. But unfortunately, in our current society, if a patient has rabies, they would be admitted to the hospital and they would spend the rest of their days until they die in isolation. So you cannot transmit. Even if you get Joanna, even if you got rabies right now, theoretically you would be able to get rabies. Because you would be admitted for the rest of your days in isolation, I would say probably in San Lazaro in a very dimly lit room, you cannot transmit it to other people because you're gonna be isolated. So yeah, theoretically yes. Not necessarily. Joanna said, "Sir, tayo po may rabies din." Girl, girl talaga, Ems ka talaga, girl. I would uh, I would leave that question to Adrian or Rainiel or maybe Cesar to answer that for you because baka masampal ka nila. How is it possible to get transmitted from the chickens? Chickens peck people, right? They have saliva as well. Isipin ko na lang po mamayang gabi. Bakit ayaw mo i-explain sa'yo ni Rachel and Christian, ni Rachel, Christian, and ni Adrian, tsaka ni Cesar sa'yo? Ayaw mo? Okay po. Yes, chicken pecks people all the time, right? There was one case before. There was one case. They thought it wouldn't be something else. And then one day the patient suffered from what was the most common. I would remember it later. I will tell you guys later. But yeah, the patient suffered from the rabies illness. A bite from a warm-blooded animal, or a bite from warm-blooded animal, and this includes again birds, can transmit can transmit rabies. All right, indicates the rules of the rabies traveled through the site of inoculation. So this is our general summary. This will be my general summary. The virions will be present in the saliva of the animal. Inoculation would be at the site of the wound, as I mentioned before. So theoretically, yes, you can get it from a chicken, and then it will travel to the nerves, specifically in the axons. And as the virus multiplies, it can reach the CNS, and therefore the effects will be evident, and you can have the clinical symptomology or clinical signs and symptoms of rabies. Now, what are the stages of rabies? We have prodrome. Again, another prodrome, sir. Ano bang kaguluhan yan, sir? Prodrome na naman. Then acute encephalitis. Acute encephalitis can appear as psychosis or even madness. And then classical brainstem encephalitis would be characterized by foaming in the mouth and then death, primarily because of respiratory distress. Remember that the brain regulates unconscious bodily activities, vital bodily activities. This includes breathing, and that's usually the death. That usually causes most deaths on patients who have rabies. Now, let's move on to explain the symptoms of foaming in the mouth. Well, nerve damage because of pharyngeal nerves. It's not because they just want to foam in the mouth. Yes, it happens, but because of the damage in the pharyngeal nerves, the salivary glands doesn't have any form of unconscious how would I say this, unconscious plugging of that one, and then they would more likely more likely release a lot of saliva. All right. Trivia: how many cases of rabies have been reported? Only one case of a patient who has recovered from rabies. And yes, yesterday whilst I was reading this, I looked at the cases in the internet, anything where I can get any information about a patient who recovered from rabies, and there was only one case, and it was actually in Japan. All right. And what is special about rabies vaccination? Well, basically, you need to have the virus in the body, and then they give you the vaccine after exposure to the virus, as opposed to your hepatitis B vaccine. They will give it to you before before doing an activity that might risk you on getting the virus. So you get the difference between the rabies vaccine virus, right? So you get the vaccine after you've been suspected of getting getting inoculated by the virus. All right? So that's the difference about the vaccination. Let's talk about toga viruses. Toga viruses have two members. There is the alpha virus and the Ruby virus. And the alpha viruses are considered the alpha toga viruses are considered as your arboviruses. And what are the important alpha viruses? Um, we have the V, the E, and the we. All right, we'll transcribe each one later. Okay, I have a specific slide for these ones, but just remember these ones for a while now. Okay, medically important Ruby viruses are your German measles, which is rubella. All right, your rubella. Okay, and the other term for German measles is your 3-day rash, which is a milder form of measles. All right, virology laboratory. We're going to talk about the virology laboratory. Just a quick recap of things that might be important for you, not just for the board exam, but also if you do have the opportunity to work in the virology laboratory. It's a wonderful opportunity. I assure you guys. You get to wear the. I know the fashion sense isn't as 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 fashionable as many of the other professions that is that is involved in healthcare, but the virology laboratory is a wonderful experience for you guys. But I think in the Philippines there is a virology laboratory. I'm not sure if it's I'm not sure, but please correct me if I'm wrong. But I think the Research Institute of Tropical Medicine is one of the places where there is a viral laboratory, virology laboratory, but it's biosafety level 4 only. I'm not sure. Correct me if I'm wrong in the chat in the comment section when you reread this when you review this video. All right? So sample for viral studies should be processed within how many hours? So it should be processed within 12 to 24 hours at refrigerated temperature or within an hour after room temperature. So please guys, if you were to become swabbers, if you were to become swabbers, you must be able to put the samples in the refrigerator within an hour. Please, please put it within an hour because you're going to put a lot of people at risk. If you become swabbers, who here has been employed as a swabber? Because I was teaching a class earlier. Um, I actually read that. I'll answer that question later because we're pretty much in a time constraint. But yeah, who here has been um who here has been employed as a swabber? Part time, full time? Anybody? Si Rona. Who's Rona? I don't see Rona. But yeah, um, I don't know if they instruct the swabbers to put it to put it in the refrigerator. Yeah, to put it in the refrigerator within an hour. It's important. Why is it important to put the samples for COVID in refrigerated within an hour? Anybody can answer me. Joanna, dadagdagan ko yung iisipin mo hanggang mamayang gabi. Bakit kailangan mong ilagay ang sample sa refrigerated temperature immediately if you swab the sample or if you collected a swab sample for COVID? What about Mera? Baka po dumami. Alright? Hindi ko alam kung anong gusto mong dumami, Joanna. Kung anong ibig mong sabihin kung bakit nadami, but we'll try to. Sir, para di makontaminate. No. Adrian, so
Properly
the
to
sample will not be
refrigerate.
denatured.
Denatured.
Um, it's pretty advanced, but it's pretty vague as well. Coronavirus, when exposed po yata, will die. Will expose to what? Will be exposed to what?
Pag na-hit po,
Para po ma-preserve. Medyo incomplete yung kay Charis. Noreen, para ma-expose sa light. Not sure. Alright, let me clear. What about Rainiel? Rainiel, hindi ka nagsalita ngayon.
Sir, cause I pretty much have the same answer din po with
Specifically
them.
what? I want to hear specifically what answer you want or you want to say.
Baka po kasi dumami yung parang yung growth po ng given uh, uh, microbes po, ganun po.
Ah,
Or like the
so I think
virus.
okay. So I think many of you are thinking in the bacteriology laboratory. You're not thinking about the virology laboratory, aren't you? You don't know what's the contents of a viral media, viral transport media. The contents are just nutrients for the contents are just nutrients for the swab samples. Remember that you're taking, yeah, it will survive longer. Yes, somebody has finally answered the correct one. Charisse, thank you so much. Right? And if the sample was left at room temperature for more than an hour, what will be the negative effects rather? What will be the negative effects to the sample? Quality control and quality assurance. Anybody? Mabilisan lang. Anybody? Noreen. Alam ko nanalo ka, Noreen, but you're still eligible to answer a question. You're ineligible to get another chance to win 100, another 5. It survives for longer at room temperature. Yes, she's correct. What would happen? What would be the negative effect to the sample if the sample was placed at room temperature for more than 1 hour? Denatured. We're talking about something. We're talking about something to do with PCR. I want you guys to think. I want you guys to think about the virology laboratory. Mamamatay po. Sino? Yung patient or yung swabber? Alisa, please be clear.
Sorry, sir.
Laloka ako, Alisa. Bakit mamamatay ka agad? Sinong mamamatay? Natakot ako.
Yung virus po, sir. Kasi po, di ba namamatay sila kapag sa heat?
Okay. So what would be the effect to the test? Quality control, quality assurance. We're talking about pre-analysis now. This is a question that will be asked of you in the board exam. Very good. Somebody answered it already. False negative. Wonderful. There would be a chance to get a false positive. Imagine the patient is positive for COVID. Okay, so I want you guys to please, if you're going to work as a swabber, please remember this. Preserve it immediately. Not because you want mamatay or what, but because you want to get an accurate result. If you don't get the accurate result, you might give the patient a false negative result, and that patient can go back to the community and spread the disease towards the other people. And I think that is one of the reasons why I know the spread of the organism in the Philippines is very prevalent, right? Because they're not teaching the swabbers to please put the sample in refrigerated temperature. A lot of the results are given false positive. Because you know why I know this? We have a lot of OFWs who are working in Saudi Arabia. They would test negative in the Philippines, and then they would go here in Saudi Arabia. They would enter Saudi Arabia. They would be tested for PCR, and they would test positive. And they would say, "I got it from the whole in the lab. I got it from your country." No, the laboratory that tested the laboratory got an inaccurate sample. Okay, or the swabber wasn't trained enough. So it has something to do with the pre-analytics. Okay? So be careful, guys, when you're doing it. So again, that's another thing that you can take from that you can take from this lecture. Not more of the not just the theoretical knowledge, or rather the tips and tricks on what questions to be asked in the board exam, but as you graduate and exit to the halls of exit to the halls of Capital Medical, you will need to be mindful of these things. I know toxic pag nagtrabaho sa hospital. I know. I know, guys. I am somebody who can attest to that. But please try to do your best to give quality results, because if a patient, especially who is going to be tested for COVID, will get a false negative result, and he will be released out of quarantine, and then he will return to the community, he will then infect more people. And remember, the more the virus replicates in the community, the more likely it will mutate into a different variant. Kaya marami tayong mga pasyente dyan sa Pilipinas nagkakaroon ng variant formation. All right? Napapansin namin kasi pupunta dito sa ano, sir, possible po ba magkamali sa RT-PCR? Actually, I've seen the machines that has been done in many of the places. I'm not sure, but Miss Matulak showed me one of the things that one of the tests that she's doing in the laboratory, and I'm pretty sure it's on the analytical basis. The only thing the only thing the technical issues would probably be running out of samples or random errors, because I've seen how they do it. It's pretty much automated, and therefore clerical error and technical error is highly unlikely. It was the most important thing is the pre-analytical errors. Okay, because technologists, because there's rarely, I don't know if they're hiring non-medical personnel to collect the samples, and they do not appreciate pre-analysis. Sometimes they would hire other healthcare professionals, and they cannot understand the quality control and quality assurance of the results, and that's the reason why I believe in, this is my personal opinion. I believe that's the reason why we're getting a lot of results in the Philippines, and we're getting a lot of cases in the Philippines, because some patients might have COVID, but they are because I've seen some of my friends, they're working in sad conditions. They only have a box with them. Sometimes there's no ice. All right, and they don't they yeah. Kasi sir, yung friend ko, positive siya RT-PCR, tapos RT-PCR ulit siya after 5 days, negative po. Well, that's dependent on the patient's, that depends on the patient's seroconversion. Not seroconversion, but rather recovery. There is what you, there's what you call a state. Maybe he already got the virus, and then he recovered, and then there's no more virus present in his body, or there's minimal amount of viral particles in his body that can be detected with PCR, and it can cause clinical conditions or symptoms, and therefore nagtest siya ng negative. Okay, I'm 5 days siya negative. Okay, so recovery is one thing to take into consideration. Remember, 14 days ang quarantine. Kaya 14 days, we want to be sure that the body is clear of any viral particles. All right? All right, let's continue because we have a couple. Nagtagal tayo doon kasi ang tagal ninyo sumagot. All right? Indicate the storage temperature for virology samples given the following storage time. 1 to 5 days, you can store the samples at refrigerated temp at 1 to 5 days. More than 6 days, you have to use a freezer, specifically an ultra raw freezer, negative 20 to negative 70 degrees Celsius. Okay, these temperature ranges fall in the realms of fresh frozen plasma. All right, and what are the preferable swabs used for viral transport? We have Dacron swabs. All right, preferable swabs. We have the Dacron swabs and the Rayon swabs. Why? Cotton can actually destroy viral particles. That's why we use Rayon and Dacron swabs. Why is calcium alginate a swab?
Sir Manuel, na-mute po kayo.
Hello? Hello, okay na? Medyo nabingi ako ng very light. Ako, na-disconnect ako. Nasipa ko pala yung ano. Sorry guys. Nasipa ko yung ano ba to? Yung USB cord nung headset. Terribly sorry. Where are we na? Balik tayo. Where are we? Protein stabilizers in VTMs. All right. So we have albumin, serum, and gelatin. The main function of gelatin, please put this in your notes. It locks in moisture. All right. Why do we need moisture? Because the cells need it. You need the moisture to for the cells within the sample to metabolize the contents of the serum and the albumin. Okay. You guys can hear me. All right. You guys can hear me clearly. Hopefully. All right. Wonderful. All right, wonderful. Now, let's move on to the next slide. We only have a couple more. Most common source of serum. Um, most common source of serum is 10% serum from gamma globulinic calf serum. Why do we need to have gamma globulinic? Because we don't want the gamma globulin, specifically antibodies, to neutralize some of the viral particles or the virally infected cells that are present in your swab. So you need a gamma globulinic calf serum. What's calf? It's basically a cow. All right. Now, examples of VTMs. Um, I'm not sure if this is a promotional video, a promotional, what do you call this? A promotional part, but Stewart's medium and Amy's medium rather, and Leibowitz Emory medium are used primarily for and HBSS are used primarily for research purposes. Okay, they're mainly used for research purposes. Stewart's medium would probably be the one that we're using for COVID-19. Eagle's medium, um, I look up, I tried to research on Eagle's medium. It's basically an egg-based serum, an egg-based combination of egg and serum. So we are much more familiar with Stewart's and the other research research related viral media. Now, HBSS stands for Hanks buffered salt solution, which is again used for research purposes. And main functions of antibiotics in viral transport media, because we want to prevent the growth of what? Viruses and bacteria and fungus, right? Because it might interfere with our tests. All right. So we want to prevent the growth of bacteria and fungus. That's why we have antibiotics in viral transport media. That's also the reason why I said sorry if I'm going to name drop you, Miss. I think it was Alyssa. Para po tumubo, ay para hindi po tumubo or yeah, AJ, sorry, it was AJ. Sorry if I have to correct you. Hindi tutubo ang bacteria sa viral culture media because there are antibiotics and antifungals. Antibacterial and antifungals. Okay? So that's why I said you were incorrect when you were answering when you answered the question earlier. Okay? Next. Next one. Malapit na guys. Don't worry. Hold on lang. Okay. Traditional antibiotics used in culture media and viral culture or viral transport media. Traditionally, we used to add penicillin and streptomycin, but nowadays we use different types of uh, broad spectrum and broad spectrum and narrow spectrum antibiotics, which is vancomycin, amphotericin, and gentamicin. All right, we'll talk about these antibiotics later on when we discuss bacteriology. Okay, what are the different ways to detect viruses? Well, There are different ways in the laboratory can detect them. And I want to summarize it for you guys. Cytology and histology, we can detect it. How, sir? We can see it using the light microscope. Yes, of course. Viral inclusion bodies can be seen in histological sections. Specifically, what? Specifically, rhabdovirus infections. What you call the nigri bodies. All right, the nigri bodies in rabies patients, particularly their brain tissues. Electron microscopy, as I've said before, is one way to directly demonstrate the viruses. Immunodiagnostic test, or what you call serology, molecular assays such as your PCR, and cell cultures, like the one that we're talking about right now. All right. Most common cytologic or histologic findings indicative of a viral infection is of course your syncytia. I've always mentioned syncytia before. It's a clump of cells that have been infected by a virus. There's also what you call viral inclusions that I mentioned a couple of seconds ago. All right. And we want to be able to detect HSV in histological sections. So how do we do that? We do the SANC test. SANC test. All right. This is to determine the histological changes of an HSV or a VSV infected cell. So what do? We take a sample from the patient. We have a cell culture with us. We add it, and we want to see what happens after infecting a particular cell, and that is after infecting the control cells. All right. So that is known as SANC test. Now, the most common staining methods for viruses is actually GEMSA and PAPS. Okay, it's GEMSA and PAPS, and the viruses that are usually detected from stains or their viral inclusions that can be stained are these things. All right, adenoviruses, CMV, parvovirus, papilloma, and molluscum contagiosum virus using, of course, your H&E stain, which is a modification of, which is the traditional form of PAPS. All right. The main advantage of viral stains is because it is what? It is not as labor intensive as isolation of viruses. All right. So it's difficult. It's useful in detecting and difficult to isolate viruses. So therefore, we would much rather see them in the sections of particular tissues rather than grow them in the. I know it's fun to grow microorganisms or rather viral particles in the laboratory, but but it's difficult, and time consuming, and it's costly. All right. What is the main advantage of electron microscopy in viral studies? You can directly observe and identify the virus. All right? You can directly observe them. What is the disadvantage of using electron microscopy in viral studies? It's labor intensive and it's impractical. Okay? Immunodiagnostic tests that are commonly employed to detect viruses: ELISA or enzyme immunoassay, and immunofluorescence, or what we call chemiluminescence. All right. What are the types of IFA that we can use? Direct, which is one step; indirect, which is two steps. Okay. We basically use labeled antibodies to cause the fluorescence. Okay, indirect fluorescence assay. So this again will not be part of our discussion, will not be taken into consideration. That's why I skipped almost all of it without discussing it further, because it's not part of my discussion. It's mainly for serology. All right. So I think that's the end of our lecture, and and yeah, that's the end of our lecture. But before I go, we still have a couple of seconds. We still have a couple of minutes. I want you guys to participate in my game show. Is that okay for everybody? Now, sino yung nag-yehe? Nabingi ako. My computer suddenly went on went on a rampage. Charing. So I'm not presenting anymore, right? I'm not presenting. Okay, so I am going to put it here so that we can see it on YouTube. This is a question. I know it's gone now. I know all questions are completed before that. All right. Ay, wait, hindi ko pala siya na-end. Wait, hang on a sec. Hindi ko pala na-save yung contest ko sa inyo. Hang on a sec. Save ko lang, ha? Uh, all right. So, I want you guys to participate for me. Ha? Sandali. Sandali lang. Sandali lang, Sir Mark. Wait lang, chill ka lang. Meron pa akong 10 minutes dito. Sir, Sir Marco, nagulat ako. Biglang nagsalita. Alright. What is it? Microbial colonies. Alright. Okay, so hang on a sec. Okay. So teach this one. I want you guys to visit this website. I created an exam for you guys. So, what are the mechanics of the contest that I have? Player versus player. All right. So, guys, if you guys have a computer right now, if you guys have a computer right now, or you have another, if you have another device with you, you can visit this website and then enter this game pin when they ask you. Okay? Mechanic. This will be just like 3 minutes. Are you guys in? Anybody? Everybody seems to be quiet. Hello? Hello? Have you guys visited the website that I gave? I need you guys to use I need you guys to use your complete name because yeah, you have to use your complete name. We can't use yeah. All right. Because this will have a prize on my birthday. So thank you but not now. Wag mo hindi ka makakakuha ng extra points. All right. So we only have 2 people. All right. Who else? Anybody still joining? Complete name, guys. Complete name because you guys that will be your scores will be listed. Your scores will be listed in an accumulative way. Okay. Aljoy, I want you to rejoin because you need to have a complete name. Anybody else? I will close the game after 5 minutes. I will close the game after 5 minutes and then we can begin. Wait lang, wala pa yung timer. Wait, i-ready ko yung timer para para fair tayo. Ayan. So, 5 minutes to enter, guys. 5 minutes to enter. Ay, 3 minutes lang pala kasi wala na pala akong time. Alright. What else? Alright. Aljoy Aquino. Alright. Anybody else who will join? Now, the mechanics of the game is in your devices, you will only have colors. You will see the choices in your screen. Okay? So that's where you have to select it. And the faster you answer the questions, the more points you get. Everyone, nobody else will join? Si Charisse, sayang oh, Charisse. Charisse, where are you? Just visit the website I posted. It's not an app. Oh, Charisse. Charisse, you only have 1 minute and 40 seconds. All right. Well, who's inside? All right, Mayra is here. All right. Okay. One more minute. 50 seconds more. All right. Before we begin. Rona. What about Rona? Are you here na ba, Rona? Oh. Alyssa is here. Okay. All right, we can start, I think. Okay. So again, your choices will be on your screen, but the answers will have to be you have to press it on your devices, okay? Clear? We can start? All right. This is mycology in colors. So hopefully you guys still remember last week's lecture because this is the question. These are set of questions that you might remember. Color of fungal elements stained with LPCB. Look at your screens, not on your device. The screen. Look at the screen, guys. Look at the screen. Okay, so only one person got it correct. LCPB stands for lactophenol cotton blue, and it will stain the fungal elements blue. All right. So again, please, look at the screen on the presentation, not on your device, because that's only the answers. All right. Unfortunately, Cesar, you can't answer in the chat. You have to answer in the exam, in the application. All right. So who got the correct answer? Myra was the only one who got the correct answer. All right, next question, guys. Color of Malassezia furfur examined by Wood's lamp. Red, blue, silver, or green? Look at the answers on the screen, not on your not on your devices. All right. There are only two people who've answered green, and we want to see who they are. Unfortunately, they didn't answer fast enough, and it was Myra who's still on the top. All right, next. All right, so Color of fungal element stained in a gram stain. Okay, your choices are red to orange, black to blue to blackish, violet, black with a reddish rosy tinge. Please enter your answers. Okay, majority of the people answered violet. Who are... Oh, Adrian answered correctly. Wonderful. Next. We don't have a lot of time. I'm terribly sorry. Apple green fluorescence is observed when a fungus is stained with what? 10% KOH plus methenamine silver, 10% KOH plus LPCB, 5% KOH with calcofluor white, 10% KOH with calcofluor white. 2 people answered correctly. Let's see who they are. It's okay, Charisse. Baltar Joanna, Aljoy Aquino. All right. This is the color of the background when a tissue section is screened for fungi using methenamine silver nitrate. Red, blue, yellow, or green. Methenamine silver. I remember I showed a lot of questions. I remember I showed you guys a lot of pictures regarding this particular slide. Yes, 2 people finally answered correctly. And I wonder who they are. Adrian and Mayra. Oh, these 2 people are contending with each other, huh? These 2 people. I love the competition. Now, what is the color of the background when a slide is stained with India ink? Black, halo white, blue, red to orange fluorescence. I remember also I showed you pictures of it, and I think a lot of people who answered halo white. There's no such thing as a halo white. All right, so everybody answered black. The color of the background is black. I think that's the last question. We're gonna see who's the winners. Unfortunately, I will only choose the top player. And who it is Adrian. Congratulations, Adrian. Your scores will accumulate for next week. Until next week. There will be another one for next week. You have to, I will take a screenshot of this one and then we'll accumulate. So as to give the other participants a chance. The prize will be announced at the end of my final lecture, which is I think December 4. I'm not sure, Sir Marco, but yeah, I think it's December 4. On my birthday. All right, that's the end of our presentation on virology, and hopefully you were able to at least get some sort of idea about what things are going to be asked of you in the exam, and not only the things or strategies that you have to on tackling the virology examinations or your microbiology examinations or board exams, but also things that I've added along the way as we've discussed. And I wish to see you guys tomorrow. Hopefully, you'll be much more prepared. And for those 3 people who have participated and have won cash prizes, 50 pesos worth of GCash, please send me your email. Okay, I remember your names, don't worry. And of course, Adrian has a copy of your names, so you can't cheat. All right, thank you. Thank you, everybody. Hope to see you tomorrow and have a nice day. Oh, huh? Oh, sige lang. Oh. Yeah, sure. All right. All right. Bye, Sir Marco.